So I'm guessing we can start soon, but it looks like Sophia's on the phone now. So has it been a good week?
Yes, so far.
Well, I need today to go and you're free. I guess it's a lot listening on Zoom all day. For was it you weren't on Monday? It was just Tuesday, Wednesday, and Thursday, wasn't it?
There was here, at least it was perfect weather for courses online.
Okay.
Hot as hell. Sorry. And
because it was so hot, or because it was so wet, or something?
Both actually. It was short rains and then very hot. Right. Perfect for air conditioning.
Okay, everyone, it seems that we are now live, so perhaps we should start. I would just welcome everybody here. Special thanks to the honored invitations from today. Vern is already here, Hanno as well. A special thanks to Quentin for his time on hosting today's sessions. So I'll just briefly introduce Quentin. Is not like he needs an introduction, but Quentin is a senior researcher at the Maze Botanic Garden in Belgium, and his most recent focus, I think you can tell a bit further in a bit, but it's on the use of information technology to research information on plants and the analysis of primary biodiversity data to understand biogeographic changes. Is also been involved in the EPES research, or the EPES initiative, and of course is a very important piece of the project Alien CSI, which is organizing this session today with us. So thank you so much for coming and for your time, and we look forward to have you here and to hear your expertise.
Well, thank you for your kind words, and I thank you for all your organization, and Joanna as well, because we really couldn't have done it without you. So I think that's a very good brief introduction to myself. I tend to be a expert in nothing actually, and but I get interested in lots of different things. But that I feel is good for citizen science, because citizen science is all about learning and things like that too. So we should kick off, but just before we get started, Alien CSI has just produced an animation with Science Seed, and Sophia is going to play that to us to sort of set the scene for what we're going to talk about today.
Science is all about the excitement of discovery. Professional scientists carry out amazing work, from gathering data to sharing new discoveries. However, individuals and communities can make contributions as volunteers to environmental and biodiversity research and monitoring, addressing real-life problems together with professionals. Contributions from citizen scientists are vital when addressing environmental challenges such as invasive alien species. These are species, such as plants and animals, that have been introduced intentionally or by mistake by humans to places outside their native region, and they can cause serious damage to biodiversity, ecosystems, the economy, and to our health and way of life. Citizen scientists are helping to address the problems of global environmental change by, for example, reporting the presence of invasive alien species to track their spread. They can also learn to identify disease-causing species and alert us. So, how can you begin to develop your own citizen science project? First, choose your topic. Work with others to come up with a question, seek to gather information to understand an environmental issue, or maybe just to provide a way for people to enjoy discovering nature and sharing their findings. Then, choose the best approach. You may prefer to work with volunteers with a specific interest, or you may aim to get everyone involved through mass participation. When you design your study, think about the best methods to answer your question, such as reporting sightings, following instructions to provide standardized counts, or running a mini experiment. Then, promote your project and accept the data from participants. Make sure you give them rapid feedback, share the results with the public, policymakers, and the scientific community. And don't forget to evaluate your project. You can also join an ongoing initiative. The Cost Action Network, Alien CSI, aims to increase understanding of invasive alien species through citizen science. You can help to stop the spread of these species by reporting any of them you may encounter. Science is for everyone, and citizen science is an engaging way to involve everyone.
You enjoyed that. I think for 2 minutes, it really encapsulates what Alien CSI and running citizen science projects is all about. So, and hopefully we'll cover a lot of the themes it brings up throughout the day. Anyway, we're already a little bit behind, and so I should go straight on and introduce our first speaker. That's Bernd Lenzner from Vienna, as you heard. And I can't quite say, yes, there you are. So Bernd I know from IPBES, where he works on scenarios and models. And I also know he's in various different projects to do with modeling and scenario building. I think he can probably tell you much more about that than I can. So I'll hand over to you, Bernd.
Great. Thanks for the quick introduction, Quentin. And hi, everyone. I'll start sharing my screen. So I guess you should see this now.
Yes, all good.
Okay. Yeah, great to be here. Nice to see you all. Some familiar faces and a lot of unfamiliar faces. I was asked to talk a bit about biases, detecting and accounting for biases in large ecological data sets, as you can see in the title here. And you see it's a bit more general because I have to say I'm not a real citizen science person. I'm not working that much with such data, but I'm working with large data. I'm a macroecologist working on large biodiversity patterns, mainly on alien species. So I will give it in this next 20 minutes like a general introduction to what kind of biases are there. I try to make a lot of connections to citizen science and point you towards studies and articles that you can then start from to get more on the specific topics that you're interested in your research. So I like to start with these kinds of comics. I like these XKCD comics, and that's kind of even though it's about machine learning, it applies a bit as well to large data sets or to big data approaches, science data approaches. So we get a bunch of data and then we need to do something with it. And the patterns, the questions we ask, the patterns we find are maybe messy, or they very much depend on how our data looks like, if we account for certain biases or how they are collected. So only stirring the pile to see when the pattern is looking right is maybe not the best option. So we need to be aware on what potential biases or shortcomings our data can have. Large data sets, I mean, you've worked with some of these already and most of them you know, but large data sets, we're in a time where we're mobilizing more and more data and more data is being accessible. So for example, if you're looking at iNaturalist, that's a screenshot from yesterday, covers about 72 million observations of about 340,000 species, which is already quite a lot. If we're looking at GBIF, this number increases even more. So in GBIF, we have about almost 2 billion occurrence records. That's a huge number of data from various sources. Or if we're looking at the map of life, we see that the spatial coverage actually looks very well. And if we're looking at these amounts of data or these spatial patterns, we would assume, that's fine, we know actually quite a lot, and we can do a lot of inferences at the global scale or at very large scales. But this data, even though it looks very nice and homogeneous across large scales, has several limitations. And there's been already a nice paper on these 7 shortfalls for large-scale biodiversity data, which was published by Joaquin Hortel and colleagues in 2015. And I'll briefly walk you through these. So the first shortfall they list is the so-called Linnean shortfall. So it's related to the species level. So we have a lot of species on Earth, but most of them we didn't even have started to describe. So we're detecting more and more new species not known to science before. And so we need to be aware that if we're making inference at large scales, that we're missing species that are not even known yet. Next bias, which you're all aware of, I guess, and I guess it has been a topic in the course, it's this so-called Wallacean or geographic distribution shortfall. So we know for many species where they occur, but we probably don't know the entire extent or the realized niche of the species. And we try to address this with these occurrence databases that we have. The Prestonian shortfall accounts for population. So what we are often lacking is species abundance. So we have occurrence data, but we don't know how many species, how large the populations are that we are counting somewhere, or how these population dynamics changes in space and time, for example, through meta-population dynamics or these kinds of things, especially at large scales. The Darwinian shortfall relates to evolution. So if we're looking at the tree of life and with respect to the traits and the evolutionary history, for many species, we actually don't know how they are evolutionary related to other species, and they are often restructuring of these evolutionary relationships depending on new findings, new methods that are developed. We have a shortfall related to functional traits or ecological functions. So there are certain traits like growth, form, and these kinds of things where we know a lot about for several species, but other traits are not as easy to access. So we often have very gappy information, especially across space and time here. Same for the abiotic niches, which relates a bit to the traits, so biotic or abiotic traits. So we actually have little information for many species where they are able to survive. And finally, we have this Eltonian shortfall, which is relating to ecological interactions. So how do species interact with each other? Do they facilitate each other? Do they compete each other? And so on. So which is especially interesting as well when we're talking about invasive species. We often don't know how alien species interact with the species in the new regions they are encountering. Of course, we're not talking about all of this, and not all of this may be important for your study or your questions, but it's just to give you an idea of what is really what kind of biases or shortfalls you can have in your data, and it's good to be aware of these things. We're going a bit more into like these recorded data or occurrence data. There's a lot of information presented by Isaac and Michael Pocock and colleagues, and they identify 4 major biases in such record data. And one, of course, is, as before said, this Wallacean shortfall. So we have a problem in spatial coverage, but as well we have problems with recording intensity, especially over time. So here you see, based on GBIF data, the recording intensity over time, or the recording of certain taxa over time. And you see this marked increase in recent years, and we have bursts of activity. So you see these spikes here, which are often related to dedicated projects or funding to mobilize specialized data. So if we're not accounting for these temporal dynamics in recording intensity, we might see trends which are actually only related to the fact that more people are going out in the field or mobilizing more data, but not true biodiversity patterns. As well, there's biases towards uneven sampling effort per visit. So the 2 main components are here: search intensity, so how many time do people, citizen scientists, spend, or normal scientists as well, spend in the field while they're out there, but as well, what sets of species are surveyed. Are we targeting our search efforts towards a specific species, or are we looking for complete species lists, and so on? And how are we collecting data? And this triangle here shows this quite nicely. So we have an increase in information content with the type of survey we are doing. So if we're recording incidental records, which we are often doing in citizen science, or if we're covering complete species lists, so we're recording everything that is there towards systematic reviews, fixed survey sites, or repeated surveys. You can see here the number of records for these different types of information changes towards the top. So we have very little repeated visit sites, but a lot of incidental records. And then, of course, we have uneven detectability across space and time, and this depends very much on different factors like the accessibility. So on top here for Great Britain, you see the recording for certain major researchers that provide data into iNaturalist, and you see here in blue, like these are only single visits, and in red you see multiple visits, and you see already that people tend to collect very much to where they are living and not very evenly distributed across space. As well, this detectability depends on available tools like field guides. I guess you have seen this very nice field guide on ladybirds designed by Helen Roy for the UK, but as well things like, do we have bat detectors so we can detect species just from their sound, and so on and so on. Yeah. These community contributed data, record data, or citizen science data have different quality, of course. And this quality can be broadly classified along a continuum from structured data, where we have data collected according to standardized protocols, so which is very much comparable across people who do these studies. We have semi-structured data, which have slightly more stringent metadata requirements. So you need to record certain information, which is the vast, and then we have unstructured data, which is collected opportunistically by many observers without like a predefined protocol or objective, but people are interested in what's happening around them, and they record a species that they encounter and put them up on a citizen science platform, for example. And these semi-structured or unstructured data are the vast majority of community contributed data that we can get. Here I have to say metadata is very important. So if we're talking about bias or accounting for bias, metadata information in such record data or community contributed data is very important. And the most important information that we need is basically what, where, when, and who of what is recorded. So what species is recorded, where was it recorded, like spatial location, we need at best coordinates. When was it recorded? So at what date or at what range of dates were the records made? And who is the person who did the recorder? So who is the observer? Here you can see I just took this from iNaturalist. So we have to assure data quality assessments in iNaturalist, you're supposed to provide these informations, which is basically the same that I reported here. And then if a lot of information is provided or the metadata is provided, then an observation can become a research grade observation, which means we have a high confidence that this record is actually good. These meta information can as well be related to recorder traits. So I highlighted this here in this red box. It's from a paper by Isaac and Pocock, which is a very nice read. So we have different recorder profiles or syndromes, they call it. So we have people who are really taxon specialists who are providing such records or citizen science information down to the casual recorder or the pan lister. And these recorder syndromes are associated with certain traits. So and the question is how relevant this information of the contents regarding to the trait and the syndrome is presented here. I'm not going into too much detail. To give you a visual interpretation of these biases in large data sets, just to show you some figures as well, and there is a nice study by Carsten Meyer and colleagues from 2016 already, where they looked at data bias in GBIF, and they tried to quantify this bias along 3 different axes, so across the taxonomy or taxonomic coverage, the spatial coverage, and the temporal coverage. And within this data cube or this information cube, we have different information provided. That's one of the central figures from their publication, and this was focused on plants. So here you can see the taxonomic coverage. We have the number of species, of accepted species that are known to science, and the number of accepted species that are covered in GBIF. And we have this for bryophyte, pteridophytes, gymnosperms, and angiosperms. And we see for many, especially bryophytes and angiosperms, we have a large gap between number of accepted species there are and the number of accepted species that are covered in GBIF. We're looking at the spatial pattern. We see here the percentage of recorded to modeled species. We see a very heterogeneous pattern. So if this is 1, it means we have the same number of recorded and modeled species in a grid cell. So we see that we have very good data in Central Europe, in Asia, in the US, or in parts of the US or South Africa. But we see that we have a lot of regions that are as well not covered very well. You have here this Moran's I value, which is a quantification of spatial autocorrelations in our data, which mean that we have values of a value of 0 means that the information is distributed very evenly across space. If the value approaches 1, it is very highly biased, and we see here a value of 0.6 is already indicating that we have quite a lot of bias. And again, the temporal coverage, we see we have an increase in the number of records over time. This is the same if we're looking at, or similar if we're looking at the geographic coverage. We have very heterogeneous coverage of species across space, and we have an increase in the records in space. And we can see here certain dips where, for example, the 2 world wars were, or something like this, where not a lot of data has been mobilized, of course, because other important things were happening in the world. And we have often this decline towards the more recent years, which is kind of a mobilization lag, which is related to the time a record has been made until it is reported and available to scientists. And again, for temporal coverage, we have as well a very heterogeneous spatial pattern of the temporal coverage of species in GBIF. We're looking a bit more on taxonomic bias, not only for plants. We see that we have very high overrepresentation of records in GBIF for specific taxa, which is not surprising. So we have a lot of information for birds, for plants, for mammals, fish, amphibians. But if we're going towards gastropods, insects, we see that we're lacking a lot of information in these data sets. We're looking at sampling bias in citizen science data. We see that we have a very strong latitudinal bias. If we're looking at space, taxonomy, and time, this is in naturalized list data for species along the California coast. We see that the observations are really heterogeneous in the space taxonomy time cube and for taxa or for phyla. So in the study by Rapacchiuolo, they looked at this sampling bias along the Californian coast, and they have very good standardized data, but as well, and compare this with the information that is provided in iNaturalist. And you see here the range completeness against the observer effort, where a dedicated observer or high observer effort is a person that observes at least 50 observations of a given phylum in the last 3 years or the last decade. And you can see here, the more active or experience or more effort is put into the sampling by an observer, the range completeness increases of the occurrence points. But at the same time, if we're looking across species, so for example, here you have 6 species that are equally abundant across the California coast, you see still that the range completeness based on the naturalized information is very heterogeneous. You can see here the 3 different species I indicate, and you see, for example, some species are more characteristic, so it's very likely that they're more easily detectable, while others are very much can be very hard to detect because they I don't know, blend in very nicely with the background, or they are less accessible to detect. So these iNaturalist data in this region suggests that there are differences in how frequently species are observed unrelated to their actual occurrence. Yeah, and this can, of course, increase the probability of false absences. So we know that we have all these biases in the data. So how can we account for this? Or how can we account for biases in such community contributed data? And I will mainly orientate and walk along the ideas of that paper by Giovanni Rapacchiolo, which is a very nice start as well for you to have a look at. And they propose like a way on how to quantify biases and where you might account for biases, and broadly follow the structure in the talk. So one idea that they propose to be first is to reverse engineer the survey structure. Reverse engineer is a bit, I don't know, I found it a bit of a complicated term, but it's basically related to filtering and aggregating observations across space and time. And it very much relates to understanding your data and the way they have been recorded and pre-processing. So, and in the first step, you actually need to specify an overall, or look at the overall structure of your data, the spatial scale, taxonomic resolution, and specify what is actually important for yourself. You need to access the spatial, temporal, and then you access the spatial, temporal, and taxonomic data and extract the ones that you are interested in. This is related to filtering and aggregation of the data to identify a subset of observations that satisfies minimum requirements. There are, for example, rules of thumb that for citizen science data, you remove sites with less than 2 visits across years, or you remove species with less than 50 observations. For GBIF or species distribution modelings, there are tools to use to account for that. And then they give a very nice example here. For certain species, you have a species list of completeness. Again, it's the California example. And if you increase the resolution, the spatial resolution, that your species list completeness increases, of course, because you include more records and you get a better coverage of the species lists you have. Another way is to borrow strength across taxa, it's called. The idea here is that the presence or the absence of a species may indicate the presence of a specific species, even if it is unrecorded. So if we have very strong pollination syndromes of these or mutualist species, the absence or the presence, for example, of a fodder plant might indicate that the very specific species is present at that site too, even though we didn't record it. As well, the absence of many common species from a survey might indicate that actually the survey effort is very low. So there's an indication for you how good such the survey and the data provided is. As well, you can look at existing reference or search lists that can help to estimate the likelihood of these false absences or focal taxa from specific observations. Often such lists are not available, but you can as well try to investigate these observations from the same location by different observers or from neighboring cells that might provide an assessment of the quality of an individual observation at an individual point in space and time. And another very important one, methods to account for observation bias is actually included into models. So we model the observation process explicitly along the questions we are investigating. So what we're doing is we include measures of variation in observer effort within statistical models. We can do this, for example, by adding spatial autocorrelation terms, by adding estimates for search time, for distance traveled during a search event, observer expertise, and so on and so on. So we actually try to explain the amount of variance that is attributed just to this observer effort and separate this out from the explained variance for other predictors. There are relevant model approaches. One is as well mixed effect modeling approaches like generalized linear mixed models or generalized additive mixed models, where we can as well include random effects that account for variability in the data influenced, for example, by groups like sampling bias or by the spatial structure or phylogenetic structure and so on. Can use occupancy modeling. There's a very nice paper that relates this very much to citizen science data, or machine learning approaches like boosted regression trees, neural networks, and so on, which is an exploration method when you're unsure which predictors are of interest in large data sets, for example. It's especially suitable for citizen science data that do not allow for classical sampling scheme, or that did not follow classical sampling schemes. As well, there is a nice preprint that I invite you to look at by Michael Pocock, which is the rapid assessment of the suitability of multi-species citizen science data sets for occupancy trend analysis, because occupancy modelings are actually very data heavy, and this is a way to assess if the data you have actually makes it suitable to run an occupancy model. Just to give you a short example, in a study in 2019, not 21, I'm sorry, we looked at alien species distributions globally for different taxa, and we wanted to assess what drives these patterns, so what are the biological, socioeconomic drivers. And for that, because we looked at the global scale, we as well needed to account for sampling effort, because you see here already the data information is quite gappy. What we did, we estimated a sampling effort for the native species for 5 of the taxonomic groups where enough data was available. And we did this by obtaining a global data set at a 1-degree grid cell resolution from GBIF, and comprised the percentage of completeness of the native species inventories for these groups based on the occurrence records and the modeled species richness. We then calculated the average completeness across grid cells for the regions that we were interested. Here you see the completeness measure for these information at the regional scale. And then we included this sampling effort as a predictor into the model. And you can see here for these 4 taxa, we see that there is always a linear positive increase or relationship between the sampling effort of alien and of alien species. So the more samples you have, the more alien or native species you have. But we account for this explicitly by including this predictor into the model. Very nice study, if you're interested in these modeling approaches, is this study here by Thomas Bird from 2014, who provides 2 tables of statistical approaches and software packages, how to implement this in R, and actually gives very nice example studies related to citizen science. Then, yeah, and then finally, and a last way to account how to account for bias is integrating standardized data together with citizen science data. So it's a combination of structured and unstructured data sets. For that, you can use point process model or so-called inhomogeneous Poisson process models. I have not a lot of experience, or basically no experience with that, but I just wanted to highlight this for you, that this exists. And there's as well a link to a very nice study, a methods and ecology study, which is very formula heavy. But the idea is here, if we have species, we see here the species intensity against the species process, so that where species occur and where we have occurrence data, and if we're using presence absence data, which is indicated or from standardized sampling strategies, we see here that we can very nicely reproduce this landscape of species presence. But if we're, for example, thin these information and only use presence only data, which we often have for especially in citizen science approaches, can see here that the occurrence or the presence of the species changes dramatically, because we're not accounting for absence information. And here we have a very small data set, like these blue triangles. They show this as with an example in Australia. If you're looking here, it's in Sydney, we have the distribution of the species, which for only from presence only data, which is very much related or clustered around Sydney. But as well, they quantify some kind of observer bias, and you can see which is related to the density of people in the region. So many people live along the coastline, and the population density decreases towards the inland. And if we include such an observer bias and run the model again, we can see here that this pattern changes dramatically with the occurrence of the species model more towards the inland. So we can assume that this pattern here is very much driven by observer bias and availability of data. To sum this up, I hope I'm not too much over time. Taking big data at face value can lead to very strong bias in estimating biodiversity metrics. So we have a very high probability to have false absences, which may lead to misestimations or misestimates of local or regional diversity and population sizes. So we need to be careful what data we use. And you really need to critically think about your data, their potential limitations, potential biases based on the method that is used, based on the people that were involved, based on the scale that you designed your study for. And then we can use these pre-processing approaches of data filtering and aggregations with respect to our research questions to take the data we are really interested in that have the quality that we need. You need to ensure that you have robust metadata to make it possible to assess these different biases, especially in observer intensity or recorder biases. And that's a very important point as well in the design or development stage of your citizen science projects, that you make it, that you really think about what kind of data you need people to enter. And then gaps and biases, of course, occur in all data sets from structured to unstructured data, but you have, depending on what data you're using, you need to account for or think about different biases there are. And luckily, we have several methods to account for such biases which occur and are readily applicable to your data, which are related to data sampling, pre-processing, or explicitly accounting for these biases through different modeling approaches, which I just briefly touched. So there's a large literature around these processes or models as well and how to implement that. With that, I want to thank you for your attention, everyone for inviting me. You can, of course, get in touch with me if you're interested in talking about these things a bit more, or if you have more specific questions. I put the slides on the G Drive. I hope you have access to that. And at the end, I as well include a list of most of the references there are, so you get a head start on looking for different interesting things related to data gaps and biases and how to account for that if you need that. Thanks.
Fantastic, Bernd. A really quick overview of all of the different things and some great sort of jumping off points for people to go and investigate themselves. Thank you. Do we have any questions? That we kind of went over our question time, but I think if there are some.
If not, I mean, for me, it's always difficult to come up with immediate questions, but feel free to drop a message if something comes up later or ask it to me directly in the chat.
And I think we'll be covering some of the topics again. I know the metadata issue is a big one for me, for instance. What we often see in citizen science projects is that we collect the metadata, we then aggregate all our data together and lose the metadata in the process. And then people then have to do that to reverse engineering to figure out what our metadata was. So so we have a lot of work to do in improving data flows within all the way from people collecting it in the field all the way to actually sort of policy relevant information and tracking all of that. But yeah. Okay, if there are no questions, then we can catch up a little bit and go straight to Hannu. Hannu is from the Senckenberg Institute, but you can better introduce yourself, Hannu, than I can, so I'll hand it over to you immediately.
Thank you, Quentin. Yeah, thank you. And yeah, hello, everybody. My name is Hannu Sebens. I'm based at Senckenberg Biodiversity and Climate Research Center in Frankfurt. And I'm a quantitative ecologist working in invasion ecology. And now I try to share my screen. So now you should be able to see it. I have to stop. I'm sorry, I have to redo it because I need to have it on another screen, otherwise it look always really strange. Sorry. So now it should be fine. Yeah, so thanks a lot also for the invitation to give me the opportunity to speak. So that's really a pleasure to present you here some ideas, although the topic I'm talking about is not so fancy at all. So it's very important, and I realized that considering data processing steps and workflows also becomes much more important also during my work. But before I go into my presentation, I would just draw the line to Bernd's talk, because he talked about biases, global biases, and also IBES, because I'm also involved in IBES. You may have heard about this, and I'm sure you heard about this. So in IBES, we try to provide a global assessment of the situation of invasive alien species. And this is really, really challenging, having all these biases and all these gaps in mind. So we can, very often we cannot really answer a simple question if we really see high numbers of hotspots of alien species richness in these areas, or is it just a sampling effect? So that's really an issue, and really makes so global assessments or a large scale assessment really problem. And it's not only about, it's one point is of course the availability of the data, which are available or not, and another one is about standardization and how the information is provided and if it's accessible. And this is more about my topic here. So what kind of data are there, and how can we work on the data to make it accessible also to others? So the title of my talk is Creating Workflows to Process, Standardize, and Integrate Citizen Science Data. But while I was working on the presentation, I realized actually the topic is much broader than that. So it's not related to just those parts here, process, standardize, integrate, and it's of course not only restricted to citizen science data. So I took the liberty to change the title to Creating Workflows to Make Your Work Transparent, Reproducible, and Fair. This is a much broader perspective, although the content of the presentation is nearly the same, but it shows you and gives you an impression that this is actually affecting your work and should affect your work and how you work. And it's not restricted just to this topic, which is covered here. So what is it all about? So in science, we heavily rely on collaboration and communication. That's obvious now, that's clear. And this is actually even more important for invasion ecology, because in invasion ecology, we deal with species which have been transported over long distances. And understanding the dynamics on one side is usually not possible when ignoring the dynamics on the other side, which are often on other continents, for example. So, and this is not feasible to conduct such research for a single person or single groups. So we really heavily rely particularly in our research discipline, on communication with people from all over the world, and also to share ideas and data. And so communication is really key to conduct such analysis, such science. But it's not only about communication, because communication is obvious. So it's clear that we have to talk to each other. But it's also about the way how we communicate, because it's not always clear that what is meant by this person is also meant by the other person. So there are many misunderstandings in terms of terminology, in terms of definitions, but also in terms of standardizations of the data. And in worst case, it may be that the data sets which are collected at different scales under different protocols and standards are not comparable at all. And also the information is not provided to allow really comparing the data and to understand what has been really recorded. You may argue that, of course, we are actually doing this. We are publishing papers, we are publishing reports, we provide information about the methodology in the method section of a paper or report. But this is actually not enough. So there are a few studies about reproducibility of studies, which includes also standardization of data sets and so on. And these are mostly estimates, but most of these estimates indicate that there is a large proportion, a larger majority of studies actually which are not reproducible. And the reason is very often that we just don't provide all the required information in the papers, in the studies, which is needed to reproduce the data in the study. Which means that the standardizations we have to do are always restricted to an individual paper or project or group or team and whatever. And this, I mean, in a paper is actually not enough space to provide all this information. And I mean, in a paper, we just we describe the methodology. We say, okay, we take this data and we analyze it in that way. But here it is all about these many, many little tiny steps which you take and all these little tiny decisions you make when processing data. For example, in my research, I'm often comparing dynamics on different countries, regions, and so on. And I really don't remember how often I had to harmonize country names or region names. So something very simple, something very basic. Of course, there are something like ISO codes, but very often these are not provided, or the region delineation deviates from the typical ISO codes. So it took me, I had to redo that again and again and again, and until I really developed my own way, my own scripts to standardize those. And this is super convenient because you can work on this over years and use and improve it, and you can redo it and reuse it. So, and all of these individual tiny steps are not, I mean, papers are not intended to provide this information, which means that overall, I guess it's much more than 70%. I probably would say that the vast majority of studies are not reproducible because of this, and therefore are not transparent. And it's also very inefficient because everybody has to do it again. So the solution is to provide the full workflow. And with workflow, it sounds a bit fancy, it's a bit weird and abstract, but it's actually something which you all do already, or you already did several times, because workflow just refers to all these data processing steps between the raw data and the final data product. And the data product can be something like a figure, or a table, or a statistic, or just a number, or whatever. So the workflow just describes all the steps in between, from data transformation from raw data to the final figure statistics, so the final data product. And when this workflow is provided in full extent in a scripting language like R, then all these steps are coded. So they are reproducible, and you can easily share it to others, and you can attach it to your paper, to your report, or whatever, and provide it accessible to others. And then it's easy to reproduce and to understand what you did actually. But let's start with the raw data. Raw data is just a very general term. It's nothing very specific. It just describes everything which you obtain and which forms the basis of your analysis. So this could be, for example, results of an experiment, or from laboratory analysis, or data you got from an online portal, or a citizen science portal, for example, or GBIF, or surveys, or so. So these are all raw data. And the raw data are not part of the workflow. So the workflow just on top of each other. The raw data provide the input for the workflow. And which means that the raw data should not be modified. So keep in mind, don't touch the raw data. You got the raw data from somewhere. It has ideally it has a version number attached, so it's clear what kind of data it is, where it's coming from. And then you have the workflow, which is taking this raw data and then modifying it. But probably ideally don't touch the raw data. So very often these are spreadsheets, and it's tempting to open it in Excel or somewhere in some software, spreadsheet software, and modify the data and save it. But this is really not reproducible. So that's far from, so that's really, and it's not so difficult to avoid this. So just save it as another document and keep the raw data separate. The advantage is that you have both workflow and raw data separated, that you can easily update the raw data, particularly these large portals like GBIF or iNaturalist or so. They are frequently updated, probably every minute, or I don't know how often, but it's easily possible to just download the next, the most recent version and rerun the whole workflow again. And also you can share the workflow to others and they can adapt it to their needs. So it's good to separate both. So the workflow, as I already said, covers all these steps of data transformation from the raw data to the final data product. And these should be implemented in open source scripting language, like R or Python or whatever. So it's important that it's an open source because it should be available also to others. And many software is licensed and is not easily accessible for others. So, I mean, something like MATLAB is very efficient, of course, very useful, but it's licensed software. And also Excel is a licensed software. And it's usually the information there, the code, the data, the output or so is stored in formats which are also not open. So that's, I mean, some of this software is really widespread like Excel, but it's not an open format. And it would be good to use only open format like a CSV, which can be easily used on all platforms and independent of license issues. And when you script, as I already said, when you script the code, the workflow and implement it in a scripting language, then you document each step directly because with document, you write it down. You write really what have you done. And this is, for example, very different when you do something in Excel. You modify the data, and I experienced this, maybe you know this very well by yourself. I looked at the table which I produced some years ago and had no clue what I did. I somehow transformed the data, several spreadsheets, copy pasted here and there, but finally I have no idea what I did. So I usually forget about the next day or so. And so that is clearly not reproducible. And this is also not very useful for yourself. So all these recommendations which I gave here is not only to provide or to make the world a better world or to come closer to open science. Of course, this is part of it, but it's also helping yourself to organize your data, organize your work, and to make the user, who's most happy about doing something in a reproducible way is usually yourself. So by reusing your own data, I'm always happy to realize when I find well-documented code for myself, which I can easily redo. The code should be user-friendly. So that means that the code, you can think of script like a text document. And when you have a text document, you have a header, you have subheaders, you have paragraphs, and you structure it to make it readable. And you should do the same for the scripts. So it's not very helpful to have a script of 1,000 lines, and then everything is somehow in there, but it's not really understandable, also for yourself not. So the script should also have a header and subheaders and a structure. You can also indicate a structure in a way that it looks like paragraphs. And of course, you should document it. So you should comment it and to really indicate what you did. Again, this is also very helpful for yourself. Believe me, that's my experience as well. And it should be organized in a logical order. Usually you have a clear sequence of steps, 1, 2, 3 or so. And then also the scripts should be ordered in that way. And ideally, that is the ideal case, you have one script which is executing all the individual steps. I will give you an example later. Of course, there are best practices how to do this and recommendation how to do this. And here, there are numerous works on this, but I would highlight 2 guidance documents published by the British Ecological Society. One is about reproducible code and the other one about data management. Because data managers is actually part of it. So it's how the data, but also the workflow is organized and presented. And these guidance documents are freely available online, and you can easily find them. And this is really very, very helpful. I highly recommend looking at those. And some basic recommendations are something like using functions. So not having the 1,000 line script, but try to separate it and try to export part of it as a function to another script. So that's more easy to read and to get an overview of what you did. Use explanatory variable names. So it's not very helpful to call the variables A, B, C, D, E, but you can also have very long names, but it's much easier if you have a meaningful name. you should provide comments. That is really helpful also for yourself. Doesn't have to be very long. I mean, ideally, you have something like you have a markdown document and a long description of it, but it's not necessary. Yeah? But just having a few words attached to each line, or to just explain what the code is actually doing, is very helpful. You can also provide readmes, and even the manual should describe in the workflow in more detail. And there are also best practices about how you share and how to provide your code, because sharing and publishing it is actually an important step here as well. So code is probably most easily, I mean, there are many ways, but these are just suggestions. Code can be easily shared using GitHub. So it's a free online platform which allows you to share your code, to upload your code, share it with others, and it also includes a version control. So you can go back to each version and re-establish the old version of your workflow. And you can also link it to other repositories like Zenodo. Zenodo is is an online repository with long-term support, where you can store all kinds of digital information. And here you can create a release on GitHub, which is directly transferred to Zenodo, where it gets a digital object identifier, DOI, associated with the workflow, but you can also include the raw data and the final data products so that everything is available together. And that is also an encouragement to you, so to make your code and also your data fair, which means that the data is, so fair is an acronym, and it refers to data principles, which are more and more adopted by institutions and researchers, and it stands for findable, accessible, interoperable, and reusable data and workflows. So with data, I mean, also workflow is part of the data processing. So it should be findable, so it should be somewhere which is accessible, which others can find. It should be accessible, so you should have, for example, a download option. So it shouldn't be private folder, it should also be the option for others to get the information. It should be interoperable. When you use open formats and open software, usually this is the case. So it should be possible to use it on different platforms, for example, and should be reusable. So others should be allowed to use the code, which you can ensure by attaching licenses which have this option. And now I would like to give you an example of how this could look like. There are, of course, thousands of different ways, and there are some gold standards, and I'm pretty far away from this gold standard still, but this example here is at least my work which comes closest to this gold standards. And I like to apologize that I'm here promoting my own work, but it's just I'm most familiar with this one, and probably this is also of interest to you because it's a workflow about standardization and integration of alien species occurrence databases. So particularly from citizen science, you have very, very heterogeneous data sets, and it's sometimes really difficult to compare both. And this workflow here is intended to, for example, standardize region names, or standardize terminologies associated with the status of a species. What means established, what means invasive, what means introduced. And also provide a way to harmonize taxonomy to make the data comparable. It is written in R, but it's not necessary to have very sophisticated R skills. You actually only need to execute one line. And if you at least accept all the default which we implemented. So this is an overview of the workflow. We don't have to go into the details here, but it just shows, or it is very useful to have such an overview, particularly when it's coming to complex workflows, to explain what's really going on here. In this case, it's actually pretty simple because the workflow consists just of more or less of 3 steps, which are executed in sequence. And it's about first preparing the data sets, standardizing the content, and then merging. So it's not very complex, but here you also see that something like, okay, you have input variables, you have output variables, also intermediate output, which can be used for cross-checking, or if you are not agreeing with the default, then you have the option to modify the defaults. And even then, it's much easier to provide this information also to others, because you can take the workflow and refer to it because it's published. And then you can say, okay, I modified it here and there, and this is my modification here by providing the workflow. So that's comparatively easy and convenient. And this is just an example here. I mean, this is, honestly, every time I open a script, I found errors and inconsistencies, and that's the same here. So don't take too serious, but as I said, also for me, it is incremental development. So I'm trying to reach, or trying to come closer to this gold standard with every project and every paper I did and every analysis. I'm still not there, but it's improving over time. And this is here one example here. So we have a script. You have ideally it's referring to one step of the workflow. It is here written as a function, and it contains a header which refers back to the workflow. So it's clear at which stage of the workflow we are. It gives a brief description. It gives some information or some documentation about this. So it's not really fancy. It's not really something very sophisticated. It's not so difficult to perform, actually, to consider. And then there's one main file, which is the file which has to be executed by the user. So, and here we have all these individual steps. So we have here the step 1 about preparing data sets, step 2, 3, 4, and 6. And these are referring to those individual steps of the workflow. So the logical order of the workflow is also reflected in the organization of the scripts. And it's comparatively easy to identify where you are at the workflow. And when you want to modify something, you can access these visual scripts easily. In this case, we published a paper to inform the scientific community. This is, of course, always depending on the workflow. Very often this is not so relevant, and you probably don't see that it could be published. It's not, of course, this is just an example to show that there's also an incentive to develop these workflows, because you can have an own publication on this. A version is provided on GitHub, and releases are provided on Zenodo together with the final data products. We also wrote a manual about this to make it accessible or easier to access. And the raw data, they are all online available too, so we just provide the links here. So this all may sound a bit complicated and a bit difficult to follow, because we have so much to consider at the beginning. But as I said, it's not really necessary, or yeah, it's not necessary to implement all of these things and to consider all of these recommended from the very beginning on. There are a few things which are more important, which help yourself to organize your work and to share your work. And it's like me, so it's also my experience that with every study, with every project you start, you improve your additional information. And now it's actually much more convenient. For example, for the Invasive reports, we have around, I don't know, 25 figures or so. And now we have everything combined in a few scripts, or actually just one script. So there's one database which is integrated from all these other ones. And then I have one script executing and then producing all the figures and tables. And this, I wouldn't be able to do this probably 5 years ago, or haven't thought about this, but it's really convenient. And a few steps which are useful to consider at the beginning are summarized here. So try to use open software and open formats, that others are able to access your files and your products. Script all steps. So it's really important to have all these steps not done in something like Excel or so. Just try to implement everything in a scripting language that is clear what you did. You can then document the individual steps. You should consider or think about structuring your scripts to make it readable. It shouldn't be only readable to the computer, but also to humans, by thinking about scripts like a text document, which can be organized and ideally provide your data. It doesn't have to be a publication, a scientific publication, but the code should be published, should be attached to the report or to the figure where it is provided, and ideally follow or provide this in compliance with the fair data principles. And with this, I would like to thank you for your attention, and I'm happy to answer any questions if you have.
That's great. Super introduction to proper open science. Do we have any questions? Oh, lots of. I'm not quite sure who was first, but well, Nadia, I can see your hands up.
Well, yeah. Hello. Thank you very much for the nice presentation. I'm sharing your your opinion on everything, really, but I always have problems with the citizen science data and the privacy law in Europe. So, for example, I have citizen science data where people mostly send their samples from home. So I just cannot provide the data publicly. And I would just like to ask for your opinion how to deal with that, because I always have problems with the journal, of course, but I cannot ignore the law, right?
Yes, no. I mean, this is an issue. So I know this also from other studies working on citizen science data, that the data providers often want to keep their rights, and it's actually not allowed to share or make use of the data. So there's no solution to this except then providing incentives to make the data available. So I mean, you have to follow and you have to accept also the decisions by the data providers, and then it's not possible. So I have also experience with, so in my experience with data curation, I had the same issue, and the problem was really is still that I'm not allowed to provide all the raw data. I'm only allowed to provide aggregated data, so you can really refer back to the individual person. But this is really very much depends on the license. I don't know, maybe Quentin, you have more to say about this.
So I have actually had to spend a lot of time reading about the law of GDPR and things. It's not as bad as people think. So, for instance, there is a clause for legitimate interest of science in data. If people have given their permission, so if people are putting things on iNaturalist and have given their permission openly, there's a sensible expectation that they wanted their data to be used. I think there's a lot of work that needs to be done that people are not thinking about when they set up things like citizen science projects, to ask very simple questions up front, so that they don't find at the end they can't do something with the data. Because just asking people, are you okay to share these data, is a very simple question, and they can say no to it, fine. But remembering to ask them first is super important. So GBIF have looked at it from a legal perspective as well, because obviously specimen data and observation data mostly have people's names associated with them. And I'm also writing papers on people in biodiversity and on disambiguation of people's names in collections. And it's quite clear from the law we are allowed to do that, and we have a legitimate interest in the scientific data on people. We do also have an ethical obligation to be careful with people's data, particularly if they're alive. And so in actual fact, that ethical obligation goes beyond what GDPR says. GDPR is actually very limited. There are things we wouldn't do with people's data that GDPR might allow, but we wouldn't want to go that far with it, for example. But yeah, that's basically it.
It's just to add on this, so it's really an issue for some, also whole databases. In some cases, whole databases are not publicly available, and there are good reasons sometimes. Yeah. So KB, for example, they have a huge database about pathogens worldwide, but it's not publicly available. And the reason is that this is actually running, and this is actually a company. So it's, yeah, they're not allowed, and they really cannot share all this information. And it's also mandate of IBES, for example, to provide incentives to make more data available, but this is a long way to go.
there's not much incentive as well if they're a company to make all their data open so that people just take it and use it. So they're not looking for ways to they want to interpret the law as strongly as possible in many cases. Any other questions? Nessa?
Yes, hi, thank you for both presentation. And I have a doubt. How can we deal with different databases? One come from expert, scientific, and one came from citizen science or GBIF. If I want, or they want to publish a species distribution model using both, can I mix up this data? And what if the expert don't want to be published or make available this data and the GBIF is available? How can I deal with the paper or mix the data sets?
That's
Is it possible?
Yeah, these are several big questions. So that's, yeah, yes and no. So that's really, I mean, it's something which many people have to deal with, not only in citizen science, but every time you have different data sets, they're very often of different quality. And then it's always a question, how can you mix it? How can you combine it? And then there's also no single answer to this. So it's really the question, what you want to show, what is your research question you want to answer, and how are the data sampled? Also citizen science data of different quality. So there's no, I mean, you can try to merge them, but when it's so different, so my approach is always to rerun the same approach using only one kind of data or the other kind of data and see how it changes. How is it affecting the overall results? So then but this has then a clear research question, in which you try to put the different data, and mentally you come up with the answer. Okay, it doesn't have any, except that it's the good fit or something. So you can pool both together. It doesn't have clear effective results, except that improving the improved. But as the database is getting more tricky because you find those, you just lump them together, and that's probably tricky. So it should reflect, you should run workflow on this and how to integrate, and then how you do this. So there's no ideal solution.
You're breaking up, Hannu, a bit. I think we caught the gist of what you were saying. Fortunately, it didn't happen during the presentation. But I think, yeah, that's a good answer, to try and keep the data separate. I would also say that keeping track of the provenance of data is difficult. DOIs help. But once you're aggregating data, then that can get rather muddled and difficult. So again, thinking of provenance before you've finished instead of after is certainly the best route to it. But it's actually an active study area of information science, is to try and figure out ways to track provenance of things.
And maybe to add what Hannu said in the beginning, it's about collaboration, communication as well. If you're using multiple data sets and you see huge disagreements, if possible, I mean, you can consult with local experts or experts of the taxonomic group or these kind of things, and get an expert idea on that there's definitely something in the data, or that makes no sense at all, or that there's a viable addition to the more robust data sets, or these kind of things. So it's not, we can't extract all information only from data. So we need to communicate, we need to consult experts as well if we're running into these kinds of problems. If possible, of course, at a global scale, that might be a bit difficult.
Okay, we better move on, because I'm getting excited by all these interesting topics and realizing that we're quite a bit behind time again, and that's going to eat in our lunch, but I'll still make sure you have lunch. Next, we have Lynn Reiserverhoeven. I've worked with Lynn in the TRIAS project here in Belgium. She's been instrumental in mobilizing citizen science data. I think you will realize once you start talking, her talk has a lot to do with the principles that Hanu has just explained, and she will be able to give a very concrete example of that in data mobilization. Over to you, Lynn.
Yeah, okay, sorry. I was just thanking you for the introduction, but I was muted, so now it's okay. I hope you can all see my screen. So the title of my talk is How to Recruit Non-Professional Taxonomic Experts in a Scientific Process. But I'm also, same like Hanu, I'm going to focus on data flows. Biodiversity data flows with a focus on invasive alien species. And I'm going to talk specifically here about mobilizing species lists, so checklists, but I also want to stress out that you can use the IDs and the workflows also, for instance, for occurrence data or sampling event data. So the bigger picture is applicable to a lot of types of data, but like Quentin says, the general idea is very similar to what Hanu just introduced to you. But so imagine in the ideal world, you want to merge data from several invasive alien species. All data is nicely recorded, is digital available, and can all be merged. So this is nice. You can use the data, you can aggregate the data, you can use them for policy reasons. And in this ideal world, all people going in the field, all taxonomic experts, they are all computer experts. They have everything digitized in nice spreadsheets. So great. This is the real world. From my experience, yeah, also there are scientists that keep everything very neatly in spreadsheets. They do their digitizing job greatly, but you also have experts that have these piles of books on their desks. So the question is how to deal with that. This is, for me, that's horror. But there is a way to deal with that, and that's great. So in the real world, you get data from everywhere, from field notes, from scans, from photographs, but also from comma-separated files, also from standardized files. And there, yeah, we need to combine this somehow. For this, in 2016, also Hanu mentioned the FAIR principles quickly. So in 2016, the FAIR principles were introduced, where the stress lies on making data findable, accessible, interoperable, and reusable. So I'm not going too deep into the FAIR principles, but the idea is that you need to be able to find the data on the web. You need to know how to access your data. You need to be able to combine your data by using vocabularies. And also your data needs a good license, and you always have to refer to the source where your data come from. So very important here in the FAIR principles are use of globally unique identifiers such as DOIs. And also, yeah, there's a lot to the FAIR principles, but I think one of the most important things is that you need to find the data and you need to publish them in a globally recognized repository. And for instance, instead of this pile of sheets, we need, for instance, something like the Global Biodiversity Information Facility, where you can publish your data openly. Here you can see, for instance, I'm not sure, you probably don't see the pointer. But maybe I don't need to use it. I'm just.
You see it okay?
You can see them. You can see.
Yeah.
Oh, yeah. Okay, great. So, for instance, here you see this data sets. The Global Registry of Introduced and Invasive Species in Belgium is actually an aggregated data set from all alien species in Belgium. It is published on GBIF. It has a DOI. You can download the data here. There is a license, the CC BY license attached to it. So by publishing your data to GBIF, you already meet the FAIR principles. I want to mention that this is one of the data sets we published in the framework of the TRIAS project. So Quinton already introduced it. I'm coming back to this a little bit further in the presentation. You don't necessarily need to publish your data to GBIF. There's also, for instance, Zenodo, where you can publish your data sets. So there are lots of repositories available. In my experiences, I use GBIF and Zenodo most of the time. So GBIF is mainly for biodiversity data, while Zenodo is more generic. But to publish your data, so starting from the raw data to standardized and fair and open data sets, we need a good workflow. And it basically is like a recipe. So we need something that tells people how to transform the data to a fair and open, the raw data to a fair and open data set. And this is what we did for the TRIAS project. So we developed a checklist recipe. So this is actually a ready to use recipe that talks through the whole process of data publication, data standardization. And it starts from the raw data. So here you see the most important figure in the paper. It starts from the raw data, which could be digital and machine readable, but it's not necessarily the case. It talks you through the different steps you need to undertake to publish your data. In our case, it was publication to GBIF. As my talk is about how to include non-professional taxonomic experts, I decided to focus on two aspects more specifically, and this is source data management first. So how do you manage your source data? It's not like this. What you see here is already a standardized file. It's a Darwin Core vernacular name extension, where you see the vernacular name of a species in column B with the language, but it's attached to a taxon ID, which is great if you're talking standardized data, but it's not really useful if you are a taxonomic expert managing your data yourself. So how do you manage your data? Like this. So, well, ideally. So you have clear column names. So you have everything nicely structured, but it's understandable for anyone working so people working on the field or people working behind their desk. So you have to keep your raw data raw. That's what Hanu also mentioned. And also important to know if you are working with non-professional taxonomic experts is that you manage your data close to the source. So I am a data publisher. Ideally, I'm not going to manage data. So data managers have to manage their data themselves. So anyone going into the field or gathering data, every time they find a new species, they should add the species to their list and they manage their data. They should not email me like, okay, I have found a new species. Could you please add this to the list? Also avoid having information sheets like this. So this is a screenshot of a Word document I received in the framework of the TRIAS project. It's not quite useful when you have these reproducible data publication workflows. What is useful is tidy data. So you use spreadsheets where every observation is raw. And in my case, in the case of checklist, that would be a species name. So each species is a row, each variable is a column, and each type of observational unit is a table. So the principle of tidy data was introduced in 2014 by Hadley Wickham. You can read a lot about it, but that's the general idea. And if you are working with people not really familiar with all these things, it is quite handy to have templates. So this is a template we used for the checklist recipe we developed. It has a few concrete examples, but the idea is that you just generate the spreadsheet yourself. They have to fill in the data and send the completed spreadsheet with the data to you. So by providing templates, you can already give them some tool to have some good data management. So keep your data raw, manage it close to the source, and use tidy data. Next, in the checklist recipe, and this is a bit all over the whole checklist recipe, is good documentation is very important. So this is why it's here below in the figure. It is related to source data management, but also to the reproducible data transformation step or data publication step. I'm not going very much into depth in this, but one thing I really want to mention is that it's really important to have rich metadata. So people reading your data, using your data, they should be able to see whether or not your data is useful, and they should understand the context. Again, when you are working with people not really familiar with data management, it's good to provide them with a template that they can complete with a few examples. That's good to keep in mind. So with respect to good documentation, it's necessary to provide rich metadata. Always think about your file management. So I didn't give an example of that, but it's like Hanno mentioned earlier as well. Don't call your file test.rmd, which I did when I was doing my PhD. I always use that example, but it's an example of horrible data management. So give them sensible names, use sensible structures, always add a README. These are just some things you need to keep in mind, which will make your life more easier and the life of everyone else using your data afterwards also much easier. So when you have structured your data is digitally available, it's machine readable, it's tidy. Then we come in the next step of the checklist recipe, that's the reproducible data transformation. What you see here is different source data files, nicely structured, computer readable, but the scientific name is first, once its Latin name, then it's taxon. I see, I put the square on the wrong place, but the column is called species. So different names for the same variable, the scientific name of a species. Yeah, which makes it a bit difficult to combine all these data sets. So how do we do that? Keep calm and use Darwin Core. When you are publishing data to GBIF, Darwin Core is the standard used there, and Darwin Core is basically a glossary of terms that could be used for the standardization of biodiversity. So it gives you lots of terms and vocabularies that you can use to standardize your data. If you standardize your data, it makes possible to combine different data sets together. So yeah, it's first, yeah. If you just use different variable column names that people use differently, you cannot combine them. Use Darwin Core or some other standards, you can combine the data sets. I just give you an example here of one of the data sets I transformed. I think this one is from the manual of alien plants. In the raw data start here is, like it has question marks, it has other signs, it has circa. So in the Darwin Core, we transform the data to the Darwin Core term event date, and all the question marks and other signs are just removed. You do lose a bit of the information, I need to say, but by standardizing like this, you can combine the data with other sources. That's the thing. Now you can do this once manually, but imagine you have updates. So each time you see here, there's a new row with new data, you have to clean everything again and again and again. It's very time consuming. So there comes reproducibility. what we did in the Trias project, and it's a bit the same thing like Hanno also introduced to you, we actually don't touch the raw data. We import them into R, and we do the data transformation using literate programming, it's called. So we write code, we transform the data using the code, and the output is a Darwin core export. The advantage is repeatability. So each time there's a new update, you can just change the code somewhat, you can rerun the code, and a Darwin core export is generated immediately. So that's a big advantage. It does require you some knowledge of R, but I will come back to that a bit later. We can help you a bit with that. But as you see, this is the script, for instance, for the manual ovarian plants I developed for the Trias project. And the thing is, it's really machine readable, but it's not really human readable. No one well, not everyone will understand what we did. But there's something called R Markdown, which allows us actually to combine code with texts. So for instance, you can see this here, we write some text, we need to integrate Darwin core, text and ID in each of the generated files, etc. What we do is really talk you through the script. And this is really handy if you are working, for instance, with field taxonomists. If they don't have a knowledge of the scripting and R Markdown, you can actually also make a website of the script. So you can just send them the link, they can read the text, see what you're doing, and in this way understand more or less what you did as a data publisher. So it's also good for yourself in about, I'm thinking about like a month. If you are rereading your script in a month, you can just read what you have done and understand it again. If you have to go to the scripts without the guiding text, it's much more difficult. So this is machine readable, and it's also human readable. One thing also we introduced in the checklist recipe is the use of GitHub. So GitHub is a very good platform for collaborative working and code sharing. So we put everything here. You can see the data in the source folder. You can find all the data in the data folder, the R code in the source folder. You can raise issues. For instance, here you see an issue related to the publication of the manual of alien plants. You can respond to that. So GitHub is very useful when it goes about collaborative working, but also version control. So each time I publish a version, I make a new pull request using GitHub. Someone reviews my code. And so every time, there's a new version available on GitHub, and you can always return to the previous version. So this is why GitHub is very useful in these reproducible workflows. I do need to mention that this is already a bit more advanced. So if you are working with non-professional taxonomic experts, this is a bit more difficult, but also like if you raise an issue, they get an email, so they get noticed, so they can follow the discussions themselves as well. And actually, this brings me to the checklist recipe we developed, and this is the last thing I want to show you. So it's very nice to have these reproducible workflows, but to have a template reproducible workflow, this is a template. This is a template reproducible workflow when you are actually a data manager. I'm talking too long. When you are a data manager trying to publish multiple data sets. So with this checklist recipe, and I'm just going to try, I'm not sure if I can, I'm just going to stop sharing my screen for a while. I'm going to share another screen. Just a minute. Okay. Okay, so now I think you are able to see the checklist recipe. So for anyone who was working with standardization of biodiversity data, and this checklist recipe is specifically developed for species checklists, you can actually use this checklist recipe to build up your own reproducible workflow. So it gives you all the tools, gives you all the explanations on how to start from your source data. So you see here the ingredients source data to finally get your dinner, the Darwin core data. So each section is clickable. It gives you a complete description on how to start. It also gives you a template. So you can download a template to upload your source data, and it tells you everything what you need to do to start from a source data to a standardized Darwin core archive. More than that, it also tells you how to publish your data to GBIF. I didn't go deeper into that topic as I got a bit time limitation. But anyone interesting to go deeper into this example of the checklist recipe can always contact me afterwards. But I think this is, my presentation is a bit more or less the same as that from Hanno, but I think my conclusion is that reproducible workflows are extremely important when you want to integrate multiple data sources, and that if you are interested in going deeper into that, you have this checklist recipe available. And there's also our paper you can consult. Also, I'm a bit advertising also from the work I did, but that's it, because that's the best I know. So I want to thank you very much for your attention, and I hope it was a bit clear to you.
No, that's fantastic, Lynn. So one thing I really like about the way Lynn does it is that is how beautiful her code looks, and especially using the markdown, it's so easy to work your way through it and figure out what's going on, because that's, even for the person who wrote it, that's difficult a few years later, even a few months later. I put a link to the Hadley Wickens paper into the chat. That tidy data paper was really a milestone paper in data science, and if you want to call yourself a data science, you need to read that paper. It's all extremely just common sense, and yet it needed somebody to write it down nevertheless. Does anyone have any questions for Lynn? Don't see any, unless there's any in the chat.
I am going to put the link to the checklist recipe. I'm not sure if you already did it. Yeah, okay.
So probably everyone wants a cup of coffee or something, and I guess we're probably still behind. Yeah, we're just 15 minutes behind. Joanna and Sophie, is that okay? We still have 15 minutes, and we eat into lunch a bit more, and maybe start the afternoon session 15 minutes later. 10 minutes, Philip.
10 minutes.
Yeah,
Okay.
yeah, I think there's no problem.
Okay. Then thank you for everyone who spoke this morning, and for those who asked questions. And I will see you back here in 10 minutes, which is, I guess, 12:40 Central European time, or thereabouts, a little bit before.
Quentin, I have to leave. I'm not joining after the break, but
Okay.
Thanks again for the invite, Joanna, as well. And
no, it was very nice to have you here, and fantastic talk as well. So
thanks. See you soon, or talk soon.
Okay.
Bye.
Bye. Hannah, are you still there?
Hi, Quentin.
Elena, how are
I'm
you?
here. I'm here. Don't worry.
I was actually seeing if Hannah was still there. I wanted a word with him, but he probably had coffee too. Still the same room. You haven't got out of the room in 2 years, it seems.
Yeah, I like it very much, so I don't want to abandon. You know, I'm very faithful.
Now that's good. There's a whole world out there you're missing, though.
No, no, no. I'm going outside later for my field work because I have to work later, so don't worry.
You work in.
But it's still easier for me attending conference at home, so not at work, because I'm sharing room with other people, so
Right.
it's more complicated.
Well, as you know, Helena, we have our first Alien CSI face-to-face meeting next week at MISA. The first.
Yeah, I know, it's a pity. I cannot attend, but
it's very, very small, but it's our first attempt at a real face-to-face meeting since the pandemic. So I'm looking forward to it, really.
It's very good. Yeah, I can imagine.
Hopefully we won't create a big outbreak of Delta variant or something, but we have to see.
No, hope not.
I can see Nadia laughing at
Hi,
me.
Nadia. Hi, Sophia. Hi, Hannah.
Hi, Elena. Good to see you.
And Vanessa, so many faces.
Hi, Elena.
Let's just wait a couple more minutes.
Hannah, I wanted to ask you about the Horizon Europe stuff, but maybe I can catch you after this if you're around really late.
Yeah, let's see. I have another meeting at 2 o'clock and I have to get some lunch. Yeah, but we can try.
5 o'clock or something like that.
Yeah, sure, yeah.
Well, no, it's 6 o'clock even. It may be late, but anyway, I'm trying to catch you and I'm busy next week, so I need to talk to you really.
Yeah, we can do so, yeah. Sure.
I suspect everyone's probably back in already and we should get going because we're already late. And okay, so now we have Elena Tucario from Italy, who you can probably introduce yourself, but she's from the University of Florence and is a specialist in invasive species of freshwater habitats. Elena, over to you.
Yeah, thank you very much. Thank you very much, Quentin. I'm trying to, share my screen. Sorry, I cannot see. Where is the file?
The big green button down at the bottom, or you're just trying to find the screen?
Yeah, but the file doesn't appear. So, I don't know why. Don't worry. No. No. I don't know why. Just for a moment.
No worries.
No, because if I can, if I can share, it show me only the screen. I don't know why. Oh, man.
Elena, you can send the presentation to me or Quentin, and then we can show it for you. You managed.
Oh, no, okay.
Perfect.
It's okay. Can you see now?
Yeah, that's good.
Okay, perfect. Okay,
So thank you very much for the opportunity to talk about aquatic environments. It's a quite different presentation to the first three. It's more qualitative approach, but just to provide you an idea about what's happening in the aquatic environments with citizen science. And if you want to have an idea of the alien species we have in the aquatic environments in Europe, I reported here some numbers. I mean, they're surely outdated a little bit because many alien species are coming every day, but just to provide an idea of how many species we have in freshwater environments and in marine environments. And you can see also the different contribution by the different taxonomic groups. So for the freshwater environments, we have many fish, followed by plants and arthropods, mainly crustaceans. But for marine environments, we have much more invertebrates, especially mollusks and crustaceans. And so remember these contributions, because this means that citizens can have some difficulties in spotting some species because these species are more difficult to be recognized by the wide public. The problem is that when you have alien species in the aquatic environments, their management is very difficult. I mean, this is a general statement that we can use also for other alien species, but this is particularly true for aquatic environments because aquatic environments are interconnected. Aquatic species can spread quite fast once in these environments, and so it can become very difficult or even impossible to manage the species. So it's quite important to spot the species. To avoid introductions or new species, but we know that this is impossible in some cases, especially for the marine environments. And so the ideal strategy should be to spot the species at the very early stage of invasion. So when you have very few population and when they are very localized. So, and in this case, citizens are very important because you know that the main problem is spotting the species when they are at the very first stage of invasion, because scientists cannot be everywhere, and so we need their help, especially if we want to avoid further spread and further impact. But we have a lot of challenges, as I told you before, in aquatic environments, because it's not easy to spot aquatic species, and because of the environment itself. This is a very recent review about published studies on citizen science and invasive species, and they are revising in this graph the different ecosystems addressed by the different articles they retrieved. They retrieved more than 120 articles on citizen science and biological invasion. And if you look at the graph, you will see that, most of the studies deal with terrestrial environments. So you have also an interesting proportion of studies dealing with marine environments, especially if the study is quite localized. But when you look at studies dealing with a much more surface, with much more study range, you can see that marine environments becomes less and less studied. And if you look at the freshwater, the situation is even more dramatic, because you will not find so many studies dealing with freshwater and citizen science. And another problem is not, environment, but also the taxonomic groups. Please remember the contribution I showed you before. And if you look at this graph, again, this is an overview of the taxonomic group spotted by the different studies the authors retrieved in this review. And you can see that if you look at the yellow part, is dealing with Europe. And you can see that plants and insects are the more targeted organisms. That is quite obvious, followed by mammals, fish, because they are more easily recognizable by people, or at least they know how a fish is. I mean, it's one of the most targeted in the aquatic environment, both marine and freshwater. And then you have the mollusks, crustaceans, algae. But if you look at tunicates, or I mean, no one look at these organisms, even if they are quite important, especially for the marine environments. But we know the citizens are helping to record the aquatic invasive species, and they are updating, they are helping the scientists to update the distribution. And I will show you that the studies are increasing in aquatic environments and the projects are increasing. So this is a good news for us. So we are receiving a lot of help from citizens in tackling biological invasions in aquatic environments. The key people involved in such kind of citizen science projects are anglers and fishermen and scuba divers. Scuba divers mainly in marine environments, but only sometimes for freshwater environments, while anglers and fishermen are very important for both environments, so freshwater and marine ones. But also boaters, tourists, students, underwater photographers, and amateurs can be involved quite easily in citizen science projects dealing with aquatic species. Looking at the different projects, there are different tools that are used, like app. I put here an example of one used app in UK, that's Aqua Invaders. Also dedicated website, and sometimes website reports also an app that people can download, like this one, Crab Watch, in northern western part of Europe. Then you can have dedicated leaflets and cards, like this one, Watch for Invasive Species in the Sea. And usually using cards and leaflets, you have an email in these cards and leaflet, and people can send information to this email. Then you have a specialized group on Facebook about fish, crustacean, at national, but also at European level. So you can follow this group, and people used to report pictures of species they observe while they are in the sea or along a river. And finally, sometimes you can use the questionnaire, so you can interview the anglers or the scuba divers about what they see. And so you can retrieve this kind of information if they are not sending any pic or any email to a dedicated website. Just to show you that also within Alien CSI, we developed this card for marine alien species. This is the Italian version. And on this card, you can see there is a contact email. So people can send what they are observing to this email. And the cards were differentiated according to the different stakeholders. So there are cards for scuba divers, for example, or cards for anglers. So, they are quite focused on the different stakeholders. And there are also different languages. So there are cards in Italian, this one, in English, in Greek, and I think also in Spanish. So, and these are dealing mainly with the marine environment. And if someone is interested, I think I can put in contact with our developers of this card. So we have different tools, but also we have different approaches of these citizen science projects. There are long-term monitoring programs. And I just read this interesting article about macroalgae in France. And since 1917, there is a group of scuba divers, performing monitoring of algae in a certain area of France. And so they have this long-term data collected by citizens on macroalgae since 1970. And they discovered along these years 14 alien species in the in the monitored area. And so this is quite important. Otherwise, sometimes projects are linked to a specific project, sorry. So to a specific project, for example, LIFE project usually launch citizen science campaign for a specific objectives or for a specific areas. And I show here the LIFE Invasive website project is a LIFE project dealing with aquatic invasive species in the Iberian Peninsula. And so they have launched citizen science also initiatives, but LIFE projects, you know, have they are not continuous project. They have a certain amount of years, usually 3 or 5 years, and after these years, the project ends. And the problem is how we can try to push further citizens and people to collect data after the project. Then you can have also bioblitz in aquatic environments. Quentin will talk much more about bioblitz, so I will not spoil anything, but just to provide an idea that during this bioblitz in the United States, the participants received a training on identification of several alien species, and through this bioblitz that lasted 2 days, they were able to detect the expansion of this invasive tunicate, so the Demnum vexillum, this one that you can see here in the picture. And so bioblitz was very useful because for the first time, they were able to detect this spread of this invasive species. Also, you can have different approaches or different aims of the project. Usually all the projects are dealing with awareness and prevention. This is a project, Aliens in the Sea, conducted in the Mediterranean area, especially in the Italian part of the Mediterranean Sea. And the aims of these projects are to increase the awareness of fishermen, divers, and boaters on the problem of marine invaders in order to avoid new introductions. But then you can have projects dealing with the early detection. And again, sorry, this is in Italian, but it's quite a meaningful example because in a Facebook group dedicated to fish and alien species, they reported a picture of the signal crayfish. Here you can see the picture. And signal crayfish is one of the most invasive crayfish we have in Europe, and is also in the Union list attached to the new EU regulation about invasive species. So member states are obliged to manage the species in the list. And so this species was believed to be present in Italy only in the northern part of the country in very restricted areas. But suddenly, last year, a picture appeared in one of these, dedicated groups on citizen science. And it was the first record of the species outside the known range in central Italy. And so using this picture, we were able to alert the competent authorities about the presence of this signal crayfish there. And you can understand the importance of this new report outside the known report at quite distant, so from the old reports. Again, using citizens, for example, in the United States, they were able to show the reports and the spread of the European green crab in the state of Washington. And also they trained the citizens to use the baited traps and to carry these monitoring activities. And later also scientists confirmed what citizens found with these monitoring activities. And again, it was quite important because the European green crab is one of the most invasive species they have along the coastline in the United States. Again, in talking about the marine environments in all the Mediterranean area, we have this project about the jellyfish. and jellyfish are quite important because they can cause a lot of damage also for, I mean, not only for the human health, but also for the economic activities. If you can think about the different blooms, stopping also touristic activities and fisheries. So it's quite important. So the Scientific Commission of the Mediterranean area launched this program, and it's a long-term monitoring program, monitoring the jellyfish blooms along the Mediterranean coasts. And you know that many alien jellyfish are coming from the Red Sea, so it's quite important so also to spot and report the blooms for coming from these alien species. And again, the marine environments, they use the citizens to spot different invasive species in marine protected areas. And if you look at the graph, it's quite interesting to see the contribution of citizens in terms of records. So you can see that the yellow part is coming from the scientific team, while the orange part is coming mostly from citizens. So the citizens are really helpful in this case because they were able to provide much more data compared to scientists, but it is quite obvious because scientists are less numerous compared to citizens. And these graphs simply show the importance of involving citizens in reporting aquatic species. And this work done in Greece about the freshwater crayfish again confirmed the importance of citizens, because the authors in this graph reported that the different source of information for different crayfish species. And you can see that, I mean, for some species, we have much more information, like the first one in blue, the noble crayfish, Astacus. But if you look at the last one, the signal crayfish again, so another, again, an invasive species, you can see that if you pull together this part coming from citizens, it's much more than the scientific literature. So again, citizens are very important. And this was confirmed also by this recent publication on Portugal and on invasive fish in Portugal. If you look at the map, the academic, data are the one in black circles. I don't know if you are able to see, but the black circles are here, here, here. While you have the unpublished data from research projects in white circle here, you can see, but you have the citizen science data with the black stars here, here, here. I don't know if you are able to see. And if you look at the map, this is quite important because citizen science data are also located where scientists were not able to have any data. And these two maps are dealing with, the first one is with the Silurus glanis, so the Welsh catfish, and the other one is with another American catfish. So you can see, especially for the Welsh catfish that is quite invasive in the Mediterranean area, is very important, this data. These data are very important because the citizens were able to spot the species where the scientists were not able to go or to retrieve them. And this is meaningful for the management, so they can start to do something with the species in another site of Portugal. Also, it's important to involve citizens because they can also assess not only the distribution, but also the impacts of the species. This is a questionnaire study conducted on the Atlantic blue crab in the Mediterranean area. And the questionnaire was, provided to fishermen, and the fishermen updated the distribution of the species, but also reported the impacts at the level of environment and for fisheries. And so in this way, we were able to understand also the impacts this species is exerting in the Mediterranean area. We can also reconstruct the invasion history of the species. Again, using volunteers and their data, and coupling the data coming from citizens together with the scientific data, I mean, scientists were able to reconstruct the invasion of several freshwater fish in northern Spain. So we're able to deal with the invasion history and to better understand the pathway. And so, I mean, when the species arrived and where and how abundant and so on. And last but not least, you can use citizens to assess the water quality in terms of biopollution, okay? So when we talk about water quality, we know that there are many citizen science programs involving citizens to measure the quality, the chemical quality of water. There are very simple programs. So the citizens can go on the river, they can sample the water, and they can measure certain parameters with a very simple kit. You can do the same with sampling the macroinvertebrates community. Usually macroinvertebrate community can provide an index of the quality of the water. And so you can do this with citizens, because while learning to sample macroinvertebrates, you learn to sample also alien microinvertebrates, and so to assess also the biodiversity, but also allodiversity, and so the quality of the streams, of the water in terms of biotic factor. And this can be done also with students. I have done with students, and they have done also with students in this study. So very young citizen science can be involved in such kind of activities. And last but not least, because I know Tim will talk much more about this, we can also involve citizens using the environmental DNA. And they have launched a program where using citizens to retrieve samples for the eDNA, so they can collect water, they can purify, and then they can send the sample to the lab for discovering which species are in the sample they collected. So, despite being a difficult, a quite difficult environment, it seems the citizens are more and more involved in detecting and reporting aquatic invaders. And so this gives us a hope, because we know that it's very difficult to work in this environment. So we absolutely need citizens. And so we know that, collaboration is the key factor. I mean, this is a general statement, I know, especially for biological invasion, but this is very important for aquatic environments, because we are not, as scientists, we have our difficulty in working in such kind of environments. We are, we are not so numerous, so we need the citizens. And seeing such kind of flourishing projects dealing with aquatic invaders and citizen science, we are very optimistic, and we hope to receive much more data and to be able to mitigate much more invasions also in the marine environment. So thank you very much for your attention.
Thank you. Really nice. Being a terrestrial biologist, I forget what happens underwater half of the time, and it's really good to see so many different examples.
Under the water.
Yeah. It hasn't got roots, I'm not interested. No, that was really nice. Has somebody got a question for Erna?
Yeah.
You can see one, I can't see one.
Okay. Yeah, I can see. Yeah, sometimes also photos are asked with fishermen, or sometimes when you interview the fishermen, you have a sort of a card with you showing the species, so they can point at the card and seeing, okay, this is the species I've seen, because we know sometimes people can mismatch species. For example, the white public here in Italy, in central Italy, sometimes mix native crayfish and native crab, because for them they are crustaceans, and they mix using the same common term for crayfish and crab. So we always show a picture. If they cannot, if they are not able to send us a picture, we show a picture saying, okay, this is the species you have seen, just to be sure they are spotting the right one.
Thank you.
Also because yeah, sometimes common name are different also in different location of the same nation. So
yeah, that's a common problem. I work with them and they usually get excited and they will say, yes, I've seen it, but and it's hard to believe them. So I wanted to know how you interact better with them. Thank you. So I
yes,
also
and
use the
yeah,
photos.
asking yes for confirmation.
Okay, yeah. I'll keep using the photos then, and I have to trust.
Yeah, photos are really useful.
Yeah, thank you.
Anyone else have a question for Elena? Oh, Hanno.
Thanks a lot, Elena, for this great overview. It's very interesting. So, based on your experience, what is your experience in the willingness, the degree of willingness of helping and contributing and sharing information by anglers and fishermen and boaters and so? Because I could imagine, okay, some are probably not really keen in helping because they would like to keep it more secret, to have all these stockings of fish or whatever. And others are maybe very enthusiastic about this. So what is your impression?
Most of them,
Yeah, it's a mix. It's a mix. I have to say it's a mix.
Mm-hmm.
Because some people, yes, want to keep secret because they, I mean, like mushroom, they have the secret place they don't want to share with the others. And also because they want to continue to fish alien species, so they don't want to share. But other people are very enthusiastic, especially if the species appear close to a protected areas or possibly threatening some native species people like very much, like newt or amphibians or something like this. So they start to share much more. Mm-hmm. So it's a mix, I have to say.
Okay, thank you.
Maybe Ovi, did you want to say something about that? Do you seem to know something about it?
That's fine. That's fine.
I think we should probably move on so we can try and keep sort of to time. Thank you, Elena. Now we have Tim Adrians, I believe. I've worked with Tim in Belgium on also the TRIAS project, the same as Lynn, but Tim has very different expertise on alien species management and using citizen science to collect data, basically, but he'll tell you all about that. So to you, Tim.
Yes, thank you, Quentin. Hello all. As Quentin says, I'm much more, I think, a man of the field and involved in many relatively concrete conservation action projects. So I won't be talking too much about data science because I'm not a data scientist. I don't even touch the ankles of people like Quentin who play with APIs, or Lynn who are masters in data publication and Darwin Core. But I know how these tools work, and I'm hoping that through giving you some examples of the work we are doing in my country, how we use these tools and how powerful they are in actually informing invasive species science, policy, and management. And as a start, I'm showing you this beautiful picture of a Mexican desert-like ecosystem. But make no mistake, this is the Belgian dune ecosystem and what it generally looks like. It has plants like these yuccas in the front and Populus deltoides, an American species. And in the back, you can see the Asian Fallopia species. So we are all hoping through our work in some way that we can help nature reserve managers to make their reserves look less like garden centers, mostly. And how can citizen science portals like this iNaturalist one or eBird, those are the ones that have vast data sets published on GBIF, or data coming from the various apps that are currently used in Europe to report selections or of specific taxa of invasive species. How can they be used to achieve this? Now, a lot has been said about the power of open and the importance of open, and I have added fair data, and Lynn's talked a lot about what is actually fair data. So I won't go too much in detail on that. You can find my PowerPoint in the course materials, and these are some very interesting papers for you to look at and to mine. But I'll mostly be focusing on citizen science data that have been published on some sort of open data platform, and there's been a lot of talk already on GBIF. And in Belgium too, we have actually achieved to publish the most important citizen science data sets on GBIF, and at the X and Y resolution, the highest one possible. And I will be giving a number of examples of how we are using those to tackle invasive species in my country. Now, as a general overarching thing, I wanted to show you this table, which we made with a bunch of people at a workshop a few years ago, trying to make some recommendations on open data. And this is just giving a snapshot of everything in terms of scientific and policy and managerial application, everything that can be done and is needed to tackle invasive species. It's going from horizon scanning over risk assessment up to risk management and making decisions on management. And I've plotted a few logos there that you will recognize throughout my talk. They're actually logos of very concrete Belgian projects that make use of citizen science data to tackle any of those activities done with those data. Now, the first one, of course, and the easiest one and the nicest one to talk about is actually that you can use a citizen science platform to be very easily and rapidly informed of new occurrences of important high impact invasive species or regulated invasive species. And then you can, through these early warnings, act accordingly and rapidly remove them from the environment. And I'm just showing you how we do it in Belgium. We have a platform called waarnemingen.be. It translates like observations.be. It's a website from the observation.org family. It publishes to GBIF, and we've built a separate portal on it, which is actually our early warning system. And it does include now, as you can see in the middle of the screen, the species of union concern, like this floating pennywort here, or this giant hogweed, or Nuttall's waterweed. And what you can do with this platform, you can subscribe to email updates as a manager, and you can get an alert in your mailbox, an overview of all the species, for instance. It's sent every day, every night, actually at midnight, you receive an email like this. If you click that link, you go to the original record. You can see the validation status, and I'm showing you an example here, a very recent one of a newly discovered population of Asclepia syriaca, a union list species. It's the milkweed, and I believe it's the host plant of the famous monarch butterfly in the States. But it is known to be highly invasive in Europe. It's on the union list and it needs to be rapidly removed. So in this case, with a little bit of early warning, and then you go into Google Maps, you look on Google Street View, you can find this population in almost real time. You can make a little history and you send it to the responsible person for removing it. Another example for birds, highly mobile organisms, where you have to react super quickly, whether it be a ruddy duck or a sacred ibis. It's basically the same principle. There's a giant community of birders, and someone will report it, even the people that are against the nasty necessity of having to kill animals for conservation. The community is so vast and so big that they will hardly be able to escape the grid, and it will be picked up and a rapid response protocol can be put in place. This is an American red squirrel, a species that seems to be establishing in my country and in the Netherlands in the last 10 years, and could even outcompete gray squirrels, apparently. Orchids in this nice heathland. There is an orchid that popped up, Aspirantes. It appears to be an artificial hybrid that's coming from horticulture doesn't even exist in nature, and it's starting to be very invasive in these prime conservation concern habitats in the Low Countries. Asian hornets, and I'll talk a little bit more about that because we've got a specific project in Belgium called Vespa Watch, and it's entirely built on open data and open citizen science platform websites. Montiac, same principle, but here, of course, the technology used to engage citizen scientists is the wildlife cameras. They're becoming increasingly popular and used also to do occupancy modeling and to determine or try and determine densities of animals based on their occurrence on wildlife camera pictures. They're a great way to involve hunters, for instance, or other target groups a bit outside the classic naturalist community. And we can use those data to quickly intervene when Montiac pop up in nature. Okay, risk management is another aspect. Of course, those data, we should not forget that even when we make decisions on how to manage the risk associated with alien species introductions, we need very good and very accurate data on where these species occur. This is especially important, for instance, when you talk about tackling at a river basin scale, but also where you have upstream populations, downstream populations, and mostly seed spread through the river. So you need to be able to use those data to unravel a little bit the source and sink dynamics before you can make a sensible management plan. And this is especially true when you're trying to do that at the scale of a country, where for all of those union list species, for instance, you need to define some objective and sensible management goal, such as here for giant hogweed, where a bunch of people in a room sat together and made a management feasibility exercise for different management strategies on how to tackle it. Giant hogweed, by the way, as you can see, is all over Belgium. So the sensible management objective we came up with was to possibly create giant hogweed-free areas somewhere in our river basins. Another aspect is management evaluation. And I'm giving you this example of a long-standing management program in my country. It's the culling of Canada geese through mold capturing, which has been going on since 2010. And here we actually use volunteer birders to count specific areas in Flanders. And I've been talking a little bit about it on day one, I think, with regards to the zero occurrences. So here we ask people to count a specific area and to also tell us for the species that they did not report to report an actual zero. And this way we can use these data in a model which you can see on the left, and with which we make time series for the different geese species present in the area. And this is hugely important information, of course, because every year the question comes up, do we have to continue this program? Where is it going? Where do we stop? Do we want to eradicate the Greater Canada geese from our region or not? And as you can see from this data, we have actually shown that since these management programs were implemented, we were able to suppress the population to about half the level of 10 years ago. Now, there's a huge work to be done, of course, before you can start using these citizen science data in such a way. And Lynn has explained how you can do this. It takes a lot of standardizing of data, because they are dispersed over various data sets. There's actually there's a mixture of professional data, museum data, institutional data, data with administrations, and the quality of which is super heterogeneous. And the citizen science data comes on top. I think we are now in Belgium in the situation where actually the citizen science data are very well structured and very rapidly published, and that actually what we see is that the professional data sets are lagging a bit behind sometimes. And of course, in order to do this, you need to adhere to standards. And Hanno's explained a lot about this. And Quentin has actually performed a lot of work to adapt Darwin Core to the Unified Invasion Framework of Blackburn et al., which you have all probably heard about. And this is actually super instrumental for being able to work with those data. And I think we will see the quality of the data on GBIF improve in the coming years more and more for invasion science. This graph I wanted to show you, it's actually what we reported to the European Commission on the data sources that have been used for the official reporting of Belgium for the regulation. Anything in purple there is citizen science data. And as you can see, since about somewhere between 2005 and 2010, the amount of citizen science data has become more and more important in this official reporting for the regulation. Well, it's coinciding with the start or the offset of the big reporting portals for citizen science in my country. So it's not a coincidence, of course. And we're also using citizen science data, such as these ones. They are the plant relevaes that are made by volunteer botanists in kilometer squares in my country. And what we can do with that is make a time series of the proportion of alien plant species in an average kilometer square in my country. And this is interesting information to say something about propagal pressure and invasion pressure on natural areas, of course. And as you can see here, this indicator is now showing an increase in the 70s of about 5 to about 10% of anything that you find in a regular square is now alien. Okay, so we know citizen science data are important. I wanted to show you this study also, which has actually tried to quantify how important citizen science data are versus professional data coming from museums. It was considering a number of invasive amphibians, such as American bullfrog and red-eared slider. And as you can see here, the specific citizen science program run in Florida, I believe, as compared to the number of records in the museum, there were much more records. They were more rapidly published. They were very important, but there were, of course, a number of biases, and that has already been explained largely. In fact, the authors of this study have concluded that the citizen science data are a very important complement to the classic museum data, because they consider mostly modern distribution data, and there was clearly a bias towards urban areas. And of course, there are other sources of bias. The most obvious one probably that some of the groups, and Ellen has talked about aquatic invaders in general, but there are other groups like termites here that are very much not accessible to the regular naturalist, so are much less reported than by professional projects. And this is nicely illustrated here. And also we are moving towards citizen science data sets that have species in them and records of species that can be photographed. And this is also clear if you look at these difficult taxa on iNaturalist. Citizen science data can be used to unravel invasion histories, and this is a nice example where the bird ban in 2005 in Europe, which was installed after an avian influenza case, actually caused monk parakeets to be introduced more in Mexico, and this was a problem for their native parrots there, because their government accidentally had already installed a well-meant ban on trapping and trading native wild parrots. So in fact, what they did was they were stimulating alien parrots to be introduced in the country and traded, and largely those parrots came from Europe. And this is something that could entirely be unraveled based on eBird data. And we're also forgetting that sometimes that we can use citizen science data to say a lot about impact of alien species on natives. And this is an example of research that we did in 2012, looking at the impact of the invasive harlequin ladybird. These graphs are showing trends in native species before and after arrival of the harlequin ladybird. And of course, you can simply make nice maps and see that the species is increasing its range in your country until every last square is actually filled. But you can do much more with these data, like you can see whether it's present in biologically valuable habitat or not, and what is the trend in that. You can also determine niche overlap with native species if you have information on the plants on which you found those ladybirds, and that you can use for risk assessment and to see which species are most at risk. And there's other things that you can learn from citizen science data, like phenological overlap. You can see on the left-hand side, these graphs are showing on the top adult ladybirds, and below is larval ladybirds. And what you see is that typically the native species have two peaks in the adult stages, with a larval stage in between when you don't find the adults. And the invasive harlequin ladybird at the end of season, clearly it still has a peak when the native species are down already. There are no aphids anymore because the sap stream in the plants is dried up at the end of season. But still we are stuck with all of these harlequin ladybirds, and they have to feed on something. So this is interesting information to say something about impacts. Okay, now let's take a look at our Vespa Watch project in Belgium and how we have actually conceived it. We have built this project for surveillance of Asian hornets onto iNaturalist. It's a citizen science project where we are trying to rapidly find any nest of Asian hornets to quickly search and destroy, basically. And you can see that we are using these data to make nice visualizations on the map. You can see how fast it's going. So in my region in Flanders, the first nest was found in 2016. And it's going hard, but we're trying to push back Asian hornets to the south and to the east, where it's coming from, northern France mostly. And this is how we conceived that project. On the left-hand side, the process starts with someone reporting a nest or an individual, taking a picture and posting it onto our website. And what is happening then is that this record is actually synchronized with an iNaturalist survey. And we use iNaturalist and its huge community of observers to do the validation. And that process is incredibly swift and very efficient. And after a record has been validated, which is in many cases sometimes instantly, but at least the same day, we sync it back to our website and we can send out an alert to a control operator. And I have called this the all-in-one approach because the website has a second module which people have to log on to, selected people. The managers, they have to register their control action. And so this way we have the whole process in one hand. We publish the data openly and we make good use of the positives of all of those citizen science platforms. And you will also see, and I'm sure Tom will talk a lot more about this, that social media like Telegram and Facebook are a very important part to keep people engaged and also a source of new records. And these are just examples of what you can do with data coming from that platform. I don't know how familiar you are with this, but this is an RShiny application. And you can just simply create a URL with a certain analysis behind it, showing data on a map. And what we have done here is we've done some, This is actually a map, a heat map that we've made, where we tried to group observations of Vespa velutina within the action radius of the species around its nest. That's generally something like 2 to 4 kilometers. And this is an interactive map, so people can log on there and can look in their region. Okay, we have a lot of observations here and the nest has not been found yet. So this is instrumental for the citizen science project itself and for the volunteer coordinators who can then organize a search action on the nest. We call the search action ivy bioblitzes. So where we go look for hunting hornets on flowering ivy in September. It's a fantastic, effective method to quickly find hornet nests. And then the fire squad can come and destroy it. And you can also show the results of the management. Of course, this is important because it's the main motivator of people contributing to your project. Quentin, do I still have time to go into the trials workflow?
Not much, but talk fast.
Okay, I'll talk fast. So we have a project in Belgium where we actually try to inform invasion policy used on citizen science data. And it's in slides. It's fantastically described in this paper that Quentin's been leading on. So I urge you to take a look. And also it created an R package and a lot of R functions that are openly available to all and that you could implement rapidly in another country. And I've posted the link here. Now what it does, it's following the whole cycle of publishing checklists, building indicators on those checklists and using occurrence data from citizen science and other sources to build risk maps that can then inform policy. And also one of the indicators that I am involved with and wanted to talk about is actually how to detect emerging alien species. This I will skip because Lynn has been talking about how to publish repeatedly checklists and how to combine checklists to registers. This is just to show you that nice risk maps can be produced. This code is all available online on the GitHub repo. You can just pick it and use it on other data sets, not a problem. And one of the cool things that we also work on is we're trying to detect actually when species are changing their range. We call them emerging species in Belgium. And the idea is really to, when we consider the invasion curve, to be able to quickly react, to have some sort of a digital e-warning, let's say. Yeah, I won't go into details of the statistics behind, but basically what we do is we make we cut loose a generalized additive model onto a time series that we take from GBEF. We account for observation efforts, or at least we try to, and we do this within and outside protected areas. And then based on the first and second derivative of that smoother, it's a method that is also used for assessing trends in farmland birds in the UK. We can say something about the emerging character, and we can actually classify a species as emerging, not emerging, or having an unclear trend. And with this, we are actually able to say something about selected alien species, such as here, the Phytolacca americana and Phytolacca acinosa, two species of Phytolacca, one coming from the States, the other one from Asia. And you can clearly see that acinosa is the rapidly increasing species of the two. But when we look at their trends within and outside natural areas of the Natura 2000 network, we can actually see that Americana is the most worrisome in terms of its impact on natural areas. Another thing I quickly wanted to mention is that within the LIFE project that has started this year in Belgium on managing invaders in river basins, we have drafted or made a workflow on the same principles and using citizen science data for drafting alert lists. So species, alien species to the country that are not yet present, but have potentially a high impact. And we've made a very relatively simple procedure that actually does the Faulkner et al. Crude climate matching based on Kupper-Geiger, and that produces maps like this, where you can choose a climate scenario, whether it be current climate or post 2070, and it simply returns to you a map of potentially climatically suitable areas for that species. Okay, that's all I wanted to show. If you have a particular interest in any of those topics, please do get in touch with any of us. All of these products are open. We're very much looking forward to them being used so that we can also improve them in the future. And I'll hand over to Quentin.
Great, thank you, Tim. Taking us back into the field. Very nice. I don't immediately see any questions, and because we're already behind, I think I would just go straight on, if that's okay, Tim.
Yes, of course, fine.
The next person up is Tom August. Tom is my go-to person if I want to know what the cutting edge of biodiversity recording is, because he's always playing around with some app or technology. But he can tell you a little bit about himself before he starts. So to you, Tom.
Thanks, Quentin. And hello, everyone. So I'm going to be talking about primarily about social media as a source of new data, but I couldn't resist to talk about some other technologies that I've been playing with that might be relevant to alien invasive species. So my name's Tom August. I work at the UK Centre for Ecology and Hydrology, and my background is in field ecology and lab ecology. I started off studying coronaviruses in bats. And for the last 10 years, it's all transitioned into a much more data science role. So basically playing with new technologies and seeing, one, how they can help us to collect better data and more of it, and two, how we can use those technologies to feed back better information to citizen scientists to help them do the things that they enjoy doing. So I'm going to hopefully, for those who haven't come across these techniques before, help to sort of raise awareness of what can be done with social media data. For those who are sort of aware of it but haven't played with it, outline what the opportunities presented by these technologies are. And for everyone, sort of tell you the lessons learned, lessons I've learned from using it, so you don't have to suffer the same issues that I've had. Okay. So I'm going to talk a bit about image classifiers and using image classifiers to extract wildlife observations in social media. I'm going to be focusing on Flickr for that example. I'm going to be talking about, and it was nice that Tim mentioned this earlier, about sort of Google Street View and how you can use Google Street View or Google Street View style imagery to collect data about invasive species. And I'm going to talk about mining text as well from social media. Let's see if I can get that guy to go out of the way. Okay. So when we talk about biological records, we're usually talking about people going out into the field and observing species in the physical world. And what I'm primarily talking about is the digital undergrowth. So collecting data and observations from the digital realm. So we're primarily talking about social media, and I've put some up here: Flickr, Instagram, Twitter, Facebook. All of these will have on them images of wildlife where someone has seen something in their garden or out on a walk, and they've put something on social media to say, oh, I've seen this, isn't this pretty, or does anyone know what this is? And so all of those can be a source of biological records, but they each have their quirks that make them more or less useful. A lot of them have restricted access via APIs. So APIs, I think you might have heard about these before. It's a programmatic way to access data on the web that stops you having to sort of trawl through manually. So, for example, something like Facebook's been burnt in the past in terms of data privacy. It's quite difficult to access data on Facebook via an API, and conversations about wildlife observations tend to be hidden away in different groups. It can be harder to access. Twitter, until very recently, you couldn't access, it was very hard to access historic data on Twitter. And they actually just made that all accessible to researchers, but still, images that are posted there are not georeferenced, tend not to be georeferenced. Less than 1% of images are. And knowing where something comes from is obviously quite important, especially with invasive alien species. Flickr is the one that I use. Flickr, about 25% of the images are geolocated, have a location. So that's great. The images tend to be of a slightly better quality because it's a forum, it's a social media platform for photographers. And a lot of modern cameras tend to capture GPS with the image. And so Flickr can pull that out, or the user can add it. So that's an advantage for Flickr. And if anyone wants to chat more about the pros and cons of different platforms, I'm happy to talk about that. I realize you can probably see this menu bar thing at the bottom, but I can't figure out how to get it to go away. Let's put it to the top instead. Okay, so I organized a hackathon about a year and a half ago, and the project I proposed was, let's try and pump data from Flickr into the PlantNet API. So PlantNet is a citizen science project, and it has an API for an image classifier. So it's a machine learning algorithm that can identify plants and images. So I had just written an R package, PlantNet, which interacts with the PlantNet API for image classification. And a friend of mine had just written the photo searcher R package for getting data from Flickr. So we brought these two together, and we created what I called an AI naturalist who is sent out on a botanical field survey of London. So all these data points are geolocated images on Flickr, which were found in a search that used the keyword flower. Okay. And then those images were then piped into the PlantNet image classifier. So you can see the scores. When I click on these little tags, you can see a score. And that score is the score that it got from the PlantNet image classifier. Every image that you give the classifier will get a species identification and a score. So you can give it a picture of a robin, as you see there, and it will still try and tell you what plant it most looked like, and it will give you a score. The score should be very low if it's not of a plant, but it will always give you a score. So you can see here already from that that sort of zoomed out view that there's lots of more images in certain places. This is Hyde Park in the center of London, which has a lot. The green tags are where it has a high score. So it's probably, the classifier is quite confident that it's got it right. The amber ones less so, and the red ones is really not confident. So that red tag there of a robin, it's saying, well, I really have no idea what this is. You get quite a lot of sort of stylistic images, like that image of the willow there. Not the sort of image that you would take if you wanted to provide an image that was really great for identifying the species. So that's our AI botanist. And my favorite image in that data set is a photograph in the National Portrait Gallery in the center of London of Van Gogh's sunflowers, correctly identified to species by the AI, which I thought was kind of cool. But it highlights one of the major problems, which is that this data set, this social media data set, is not created for the purpose of biological records. And so we have stuff in there which is just not appropriate, such as artistic representations of flowers. We also see a big spatial bias. So after that hackathon, we went on to write a paper, and we looked at two areas of the UK, London in the center there, which you've kind of already seen on an interactive map, and on the right-hand side, the Peak District, which is a big rural area of the UK. You can see that within these areas, there's big clusters of images. It's spatially biased. You've got Kew Gardens in London, that's A. B is the Chelsea Flower Show, an annual horticultural show. And C is Chatsworth House, a kind of public attraction, a big stately home, which also has a flower show. So there's lots of spatial bias, but we're not, we know there's spatial bias in all citizen science pretty much. So that's not something new, but it does seem quite extreme in this data set. If you look at the distribution of those classification scores, that's what's shown on these two histograms here. Anything that's above sort of 0.8 on that right-hand end, the classifier is fairly confident it got it right. But anything towards that bottom end, I'd say anything below sort of 0.4, we need to certainly question. And so you can see that there's a large number of images which have these very low scores. And when you look at those, you can see there's an obvious mismatch between the images that we're trying to classify and the images the classifier is designed to classify. So these two images at the bottom are classic images that we might get from Flickr that are of flowers, and it's a flower bed. So which one species are you expecting the classifier to classify from those? Well, it's an unfair question. There's lots of species there, and the classifier just gets confused. And so the reason, one of the main reasons we get these poor performance is because it's mismatch between the images we're presenting to classify and what the classifier has been designed to classify. If we dig into that a bit more deeply, you find that naturally occurring species tend to be better classified, native species tend to be better classified, and where the images of a single plant, such as the flower head of a single plant species, it performs best. So we really need to think about what is the classifier designed to do when you're applying it in this sort of situation. I want to pick up a few, there's a whole list of questions that sort of came out of this. The underlying ones are ones that I haven't really spoken about that's worth spending a little bit of time thinking about. So the first is about filtering the images. So this search was done on images on Flickr which have the word flower somewhere. Now, you might think that, well, if you have flower, then you're going to get flower beds, which are displays of multiple species. So maybe we could have used a search term which excluded flower bed as a term. There's lots of things that you could do additionally to help to filter those images down before they come into your classifier. It's also fair to say that by using the search term flower, we got not many images of trees. So you could also might want to do multiple searches using different search terms. The second question I've underlined here is about propagating uncertainty. Now, when you get a traditional citizen science observation, it tends to have a species determination, and that is binary. It is that species or not. When we use AI, the classifier gives it a score, usually between 0 and 1. And so that is an indication of uncertainty. And there's some thinking to be done about how we can incorporate that uncertainty into our models of spatial distribution or trends over time. And finally, it sounds like you've already heard about documenting data sets and sharing data sets. How do we properly document an AI created data set? What's the metadata that needs to go with that? And I'd suggest you would need to have DOI for the model that's being used. You'd want to have the classification scores. Ideally, you'd want that model to be open, so that model could be tested on different data sets. And there's a lot to be thought about in terms of what metadata is required to make the most use of these data sets going into the future. Okay. So that is quite a passive citizen science approach, right? So it's, we're using data that's created by members of the public, but they're not involved in the scientific process. So some people might say that's not really a citizen science approach. This next one is taking on the Google Street View idea. So the idea was Google Street View is effectively a transect through the landscape that's repeated over time. And so that can be very useful for monitoring invasive species along the roadside and monitoring how they change over time. And since a number of alien invasive species will spread along road networks, it's quite well placed survey. The problem is I approached Google and they weren't really interested in sharing all their Google Street View data with me. There are restrictions around the use of their data and their terms and conditions, which kind of made it kind of impractical. So we built our own system. So you can see the little white box on top of this car. Built this hardware and we sent it along roads, and we created training data sets by having ecologists review the images collected and draw little boxes around where they saw the species of interest. And we've created an image classifier for 2 invasive species, Himalayan balsam and Japanese knotweed. And we've also looked at ragwort and ash trees, which people are interested in. So this is sort of in early days, but we've sort of had our proof of concept. It seems to be working well, and we're looking at scaling this up now. Also, in terms of testing on a larger area, but also putting the camera onto trains so we can monitor the vegetation along railway lines. And it could also be applied on boats, going up and down canals and rivers to monitor river vegetation. So that is a sort of professional survey. You can imagine it's also being done by citizen scientists as well. So Mapillary, this map on the right-hand side here, these green routes show you dash cam footage collected by members of the public. So it's kind of like an open version of Google Street View. Okay, so it's kind of like OpenStreetMaps is to the Ordnance Survey map, or your national surveys of roads, is what Mapillary is to Google Street View. The main issue with this at the moment is that the images are primarily forward facing, because that's the way your dash cam normally faces. So we really want the camera to face sideways. The one cool thing is you do get data along rivers. So this image bottom left, this is actually only a couple of 100 meters from my house on the Thames. And this has 360 images every sort of 5 meters along the Thames for this stretch. And these would be good enough to be able to ID the vegetation. So I think there's definitely more potential here for looking at these citizen-created data sets of linear features, be they rivers or roads, and then applying these computer vision algorithms to try and identify the alien invasive species. It's also worth saying that the ambition here in our project is not to replace traditional surveys, but to help to target them. So you can run an AI across all this data, and then it can pick out hotspots of where it thinks alien invasive species are. And then humans can then go in and review the images, the data from those areas, or go out to those areas to investigate in more detail. I want to talk now about text and text in social media. So Twitter is a great source of data, and this is just a little study, and actually better studies have been done elsewhere. But this is a little study that we did looking at mentions of the harlequin ladybird, and we've showed here in the graph on the right hand side that kind of dark blue olive, that olive green line. That's data coming into iRecord, which is a UK citizen science platform. So you can see the sort of peak in the summer of harlequins, and drops down in winter, and then a sort of second peak as we go into the second summer. The blue line are tweets that mention harlequin ladybird, and you can see that it does pick up the sort of main peak and trough, but it's also more peaky, I would say. So it seems that in this case, at least, Twitter is kind of more responsive to changes in abundance. And so when the abundance changes, you get perhaps people saying, oh, I'm seeing more harlequin ladybirds. And in that way, it might lend itself to early warnings of changes in wildlife, such as perhaps alien invasive species. We thought there was a tentative signal that Twitter was a few days before iRecord in being able to detect these changes, but you need a much larger study to look into that, and much larger studies have been done. So if you search literature, you'll find many more. I've recently had a undergraduate student who did their final year project on sentiment analysis, and this is using Reddit. So Reddit is another social media platform. The good thing about Reddit is it's designed in a very open way. So it has an open API, so you can search any bit of Reddit that you want to. And there's also a service which allows you to query the entire archive of Reddit. So it's very open, it's very easy to get data, which is great. It's a forum, so people are discussing topics there. And the particular interest here was in public sentiment towards bats. So my research background is in bats, and I was interested in how COVID-19 affected the public's perception of bats. There was a lot of, in the early days of the outbreak, there's a lot of kind of misconceptions about the disease emerging through eating bat soup and that sort of thing. And that resulted in kind of negative sentiment towards bats, calls for culls of bats and those sorts of things. So I was interested in whether we could pick up that signal in Reddit, and if we could understand what was driving that change in sentiment and how it changed over time. So this is what Svetoslav looked at. Bottom left corner, these two graphs, what you're seeing on the y-axis is sentiment. So we can give words a sentiment score. So negative, so something like hate would have a very negative score, and a word like love would have a very positive score. And then you could have something like not in front of a word will switch its sign. So not good, we'll take the score of good and change it from positive to negative. And then there's amplifiers like very or not very, which sort of modify their scores. So sentiment analysis is a really interesting literature to look into. It's mainly designed for things like product reviews on Amazon. It's not really applied in the biodiversity context. So so this is quite a novel application. But so here we've got two different sentiment metrics. There's a whole bunch that Svetoslav looked at, but you can see that they kind of pootle along until halfway through the graph, and then they drop down. This is when the first, when COVID sort of, the outbreak first occurred, reaches a trough, and then it comes back to recover. What we find is across all of these sentiment scores, it never actually comes back to that green line, which is the average pre-outbreak. So there is this kind of permanent damage done to the sentiment around bats on Reddit, but it does have this sort of drop, and then it does recover most of the way. So that's sentiment. Another thing you can look at is something called n-gram analysis. That's kind of word association. So you can look at what words are associated with the word bat. And you can look at bigrams, so like 2-word combinations. We can look at trigrams, which is 3-word combinations. And what these are doing is helping to understand the context in which the term is being discussed. And that's what you're seeing on this image on the right-hand side. So the top are the words associated with bat pre-pandemic, so kind of in the kind of control period, as it were. And there's no real term which is standing out there. The bottom is in that trough region. So when we saw that really negative sentiment, and so you can see eat comes out really big, virus, soup. So you can see those sorts of, eating bats was not the source of the outbreak, and eating bat soup was not the source. So you can see that misinformation is clearly playing a role in this negative sentiment. It's worth saying that there's a lot of things to consider here in terms of filtering the data that you're getting out. So we searched for bats, but we did put a lot of effort on stuff, but a lot of effort into filtering out if people talking about Batman or baseball and things like that. Again, a very noisy data set, but a lot of data there. So once we'd done, even though we had to do a lot of cleaning, there was still a lot of data left over to play with. And I think this tool could be useful, I think, in the alien invasive species context as well, because you could look at how sentiment towards species changes over time when an emergence of an invasive alien species occurs. You can look at word associations, and if done in real time, you can then change your messaging to help to combat misinformation or misunderstandings and those sorts of things. So that's me, finishing pretty much on time. So computer vision methods are great for lowering the barrier to species identification. They create these probabilistic classifications, but when we're using social media, we need to really think about the noise in that data. Ecology is the term given to using these online data sets, not collected for biodiversity recording, but using them nonetheless for understanding biodiversity. So there's new sources of data, but they have their own biases. And these linguistic analyses give us a kind of interesting new tool to understand changing sentiment and to understand what's driving sentiment. And I'm just going to leave it there. So thank you very much.
That's fantastic, Tom. I hope that's given everyone else as many ideas as it gave me. So, does anyone have some questions for Tom? Arjo?
Yeah, sorry, it's me again having a question.
That's not a problem at all.
Yeah, Thomas is there. Yeah. Thanks very much for a brilliant talk. Thank you. I was just wondering, I learned during my PhD that the media work has, so with media work of your citizen science project, such as a Harlequin project, you might create your own bias in the data. So the amount of records you get. Did you check with your Twitter analysis and the iRecord, the graph you've showed, if that has something to do with your own communication strategy?
So that definitely has an impact. So if you have a news story on the national breakfast news, then you'll get a big spike in records for sure. And we have seen that exactly with Harlequin Ladybird in the UK Ladybird survey. I think the overall, there was a smoother applied to that data, and so the kind of humps that they were related to the phenology of the species. But you're absolutely right that you need to consider how media might impact in the shorter term, especially if you're looking at like shorter temporal periods.
Mm-hmm. Okay, thanks.
All needs to be documented in your metadata, I think, as well, doesn't it? Basically. Anyone else got a question? I think probably
I have a question. Sorry.
Go ahead.
I'm just wondering if you have experience in extracting information from Instagram as well, particularly like pictures and georeferences, if you have species on the pictures.
So I missed, I think, a keyword in that.
So if you have experience with Instagram, extracting information from Instagram.
Yeah, no, I haven't. I think I briefly looked into whether they had an open accessible API, and I didn't find anything. Okay. Instagram is owned by Facebook, am I right?
Yes, I think
so I'm imagining it's probably fairly locked down. I don't know if anyone else has experience. No.
Okay.
And I haven't used AI or machine learning, but I have been looking a lot at iNaturalist pictures, and there are just so many and so much information packed into those pictures of all different sorts of things. So we have a paper in Press which is on looking at how people handle bats and whether they're using PPE when handling bats, for instance, because you get pictures of people holding bats with their bare hands and no mask on, which is not good for the bat and it's not good for the people either. But I've talked to Tom before about pollinators and plants that they're sitting on. There's just enormous amounts of information hidden away in all those pictures about lots of different aspects of ecology.
Yeah, and it's great to see how these AI image techniques are developing, and having something like iNaturalist, this big repository of images, a lot of them with licenses that allow you to use them for these sorts of things. It's a great repository to be able to go back to as these techniques improve, and we can do more with those images to be able to go back and look at answer questions like Quentin's.
Yeah. Okay, I guess if there's no other questions, people probably want their lunch. We meet back here at 3 o'clock Central European time. I guess that's 2 o'clock Portugal time, UK time. Thank you everybody for this second session. It's been fantastic. I've really enjoyed it. I hope you have. And I should mention just before we do so, I'm giving a talk on BioBlitzes when we get back, and then we're combining with the Alien CSI Coding Club, where we run that every Friday at 4 o'clock Central European time. Anyone can join that if they would like to, of the people participating in the training workshop. Just send me an email. And we're going to look at OpenRefine. So I hope you didn't do OpenRefine sometime during this week. But I have one of my guys from my team who is going to explain Open Refine to us. And if you still want to hang around after that, then we can do some hands-on work with using Open Refine to clean data sets. Okay, so see you in an hour's time, or just less than an hour.
See you.
Bye, see you.
And it looks like people are coming back online again now. See people appearing slowly.
Hi, Quentin. Hi, all.
Hi.
Welcome back.
I wonder if we can just get started.
I think probably we will need to wait 3 minutes or
Okay,
so.
I don't know how many people are supposed to be here, so we can wait. It's absolutely fine.
But usually 5 minutes and then we start.
I'm not sure we'll go to 6. Well, it's 5:30 or 5:15, 6:15. We'll just see how it goes. And
Exactly.
well, you have had a very long week, so I don't think you'll mind stopping a bit early.
Especially on a Friday afternoon, right?
Oh, you see I'm on message?
Yeah.
Nigel, you were asking me about the PlantNet thing. The PlantNet people themselves were also quite approachable too. So.
Oh, that is great, because if there is like a package where you just can feed in pictures and they might automatically like create a project or so in PlantNet, would be perfect.
Yeah, I think they let you do 1,000 a day or something like this for free. So if you have 1,000 images and you want to pump them through, it's fine. If you want to do bigger things than 1,000 a day, then you have to talk to them. But
yeah,
still pretty good.
That would be perfect to for the interaction project to just get the plants identified. Yeah.
Yeah. Although I was thinking in Darwin Core, I don't think there's a place for putting the probability that it is that plant or the identifications, but I think it's been talked about actually.
Yeah, I was thinking about if if the score, the probability score is too low, maybe you can even try to upload it again to let the user from my naturalist verify them, or if there are only a few hundred pictures, you might be able to do it yourself or with a PhD student.
Yeah, yeah. But often it is, when you look at the ones with a low score, they are actually pretty terrible pictures or have completely the wrong things, and even a human being finds it problematic. So
yeah. Yeah, I would need to have a look at the TensorFlow package that was presented here as well. So maybe that is a good way to classify the images before putting them into PlantNet. So I will think about that.
Good ideas.
And you can use TensorFlow through Google Colab to make it faster.
Through what? Sorry?
You can use TensorFlow through Google Colab.
Yeah.
Because it is much faster to process.
So we did Jupyter notebooks in our coding club, but I didn't do it through Google Colabs. I used the Anaconda version, but yeah, Google Colabs is quite cool too. It's not so good for if you want to get external files in there and things, so let's hope to get tricks around that.
Especially with TensorFlow 2, it makes it harder to
okay. So Sophia, do you think we should start now?
I think so. We waited the additional 5 minutes, so.
Okay, so yeah. I think I'm scheduled actually to talk about BioBlitzes for the next 20 minutes, 30 minutes. Our normal coding club session is at 4 o'clock my time, which is 3 o'clock your time, some of your times. And so I'm aware to try and push my session to about an hour, talking about BioBlitzes. I don't want to talk for an hour. I want to have a discussion about BioBlitzes and how you could think that you could incorporate BioBlitzes as a method into some of the things you've learned this week. Because I'm meant to be organizing some sort of practical session for you, but that practical session will be on OpenRefine, and we can think about BioBlitzes as just one of the methods you can use for collecting data from citizen scientists. So anyway, let me go to my presentation, and please just blurt out questions to me. And if anyone's if anyone has bad bandwidth, please do keep your cameras off. But if you don't feel too shy, keep your cameras on, and I can see you and I can see reactions, and it knows I'm actually talking to some people. That's really useful for the speakers. So, okay, share screen. Okay, so hopefully I expand that so I can see you all too. Now I guess you see my presentation, do you? Do I see some nods? Yeah, okay. So this is about BioBlitzes. So BioBlitzes means lots of things to different people, but I want to let you think of it as a method, a tool that you can use maybe to collect data, but for other things within your work in biodiversity informatics and in biodiversity in general, in ecology even. So, click there. This talk is kind of a summary of work we've been doing in Alien CSI with these people here. It started off with a short-term scientific mission that Yolanda Rocha, I don't think that's how you spell her surname, but anyway, did at MISA, but we've since taken it forward, and we've been having regular meetings throughout the pandemic and building on this, and different people have done different bits of work on it. And in actual fact, we have a workshop next week where we are going to hopefully finish the paper and have it all done and to be submitted in the next month or two, maybe 3, and that's how quickly we get on. And I guess it even goes back further than that, is that we had a BioBlitz in Akrotiri in Cyprus at, I think it was our first management committee meeting of the cost action. where, well, I'll tell you a bit more of that later. So for those of you who are not German speakers, Blitz means lightning. Nadia will correct me if I'm wrong. And basically it's about it being fast, short, sharp shock. The Americans use Blitz a lot for when they're cleaning their house at the weekend to try and clean it up very quickly. But a BioBlitz is about doing things in that sort of short, sharp shock way. When we first started sort of scoping out this paper, we realized that many people also use BioBlitz for all sorts of things. Basically using the term Blitz as something kind of intensive. And we had to define it to make it easier to do our literature searches and things like this. And this is the definition we came up with that was based on other definitions that other people had published. So BioBlitz is a rapid assessment. So that's the whole Blitz part of it. It's obviously about biodiversity. That's what the BioBlitz on it. We made it linked to a specific geographic area. So there are some big globalish ones like the Nature City Challenge, but those are still specific areas because they're bounded by city boundaries. And so even though it's a global event, each city has its own BioBlitz within that on the same weekend. And we also stipulate that it's about multiple people engaging. Occasionally you get people online naturalists who say they're BioBlitzing their area and it's just them going out at the weekend and trying to get as many taxa as they possibly can, which is fine. It's a good thing to do and people enjoy doing it, but that's not what we wanted to examine in terms of BioBlitz as a method. So it really was about an event where multiple people are involved. And those people are not necessarily citizen scientists at all. In actual fact, if you go back to the first event that was held in, I think in Washington, DC in the United States, when you look at the participant list, the vast majority of people on that list were people engaged in some sort of biodiversity research. They were from the museum, from local nature conservation organizations, and things like that. So they were actually professional people who were volunteering at the weekend to go to a nature reserve and just try and identify as many things as they possibly could. So BioBlitz is not necessarily about traditional citizen scientists, but it can be, but it's also a lot of the time about experts getting involved and volunteering their own free time. So if you go and take all of the, well, you probably don't, I know I'm naturalist quite as in and out as I do, but iNaturalists can have projects, and some of those projects are tagged with the name BioBlitz. So they have the name, like we did one called the Akrotiri BioBlitz, for instance. And if you, each project has a description of that project, and if you take all of the words of that description and make a word cloud of them, that is what you see in front of you there. So this is just a very crude way to get some idea of what other people think their BioBlitz is about. So clearly biodiversity is quite big on there. Event is quite big on there. So clearly having that sort of time limited aspect to it. Observations is on there. So that's about collecting data on particular organisms. But there's some other interesting words that are relevant here. You see the word park down here. You also see park services somewhere on here. There's national park. There's garden there, which is in French as it happens, but there's also, some of these are in Spanish because there's quite a lot, mostly from South America rather than Spain. So it also gives that sense of place that a BioBlitz has in there as well. And of course you have nature in there, find, discover, and learn is quite prominent too. And I'll get onto that in a bit. So this is kind of like our just sort of scoping for understanding a bit about what other people think about BioBlitzes and what we think about BioBlitzes. And right at the beginning, I think you can probably think of some more reasons why you might hold a BioBlitz, but we particularly investigated these 5 things here. So Creating an inventory. So just, I have a nature reserve. I don't have enough staff to go out and spend their afternoons and looking at what species exist here. I just want a full list of all the things in my nature reserve. How do I get that done? I'll organize a BioBlitz. Discovering new species. So those might be looking for rare species, for instance, or they might be looking for invasive species. The engagement of the public, and that has many different threads to it. Improving the knowledge of participants, so that's that learning aspect. And the promotion of an organization. So organizations like my own Botanic Garden, we run events to promote ourselves to some extent. I think we don't do it quite so explicitly, but I'll get onto that in a bit. So the first thing we did was, well, Yolanda did in actual fact, was do a literature search where she was looking for published examples where people have published their information about a BioBlitz. And then a bunch of us went through and we scored each of those publications for how, after reading them, we thought that those organizers, or at least the authors of the publication, felt about the aims of their BioBlitzes in terms of the 5 different things that we listed. So if they thought, if the paper was 90% about teaching people in the BioBlitz, then we would have given that a score of 5. And if they didn't mention creating a species list at all, then that would get a rank of 1. So 5 is the most important for the people who organize that BioBlitz, basically. And so what we found out, it's rather scattered, but you can see on the left-hand side, which is the least important, promotion of organization was not considered in these papers very important at all. And on the right-hand side, did I say the right-hand side before? I mean, the left-hand side is promotion of organization, not important. On the right-hand side, creating a species inventory is very important. But you can also see the pink public engagement is quite important to people too. New species is sort of scattered throughout there, and also learning as well. So no clear winners, except maybe a clear loser of promotion of an organization. And maybe inventory, creating an inventory is probably the winner if there is one. So where are BioBlitzes? Well, we mapped the ones that came from the literature. I suspect they were biased towards the English literature anyway, but they were mostly sort of North American and European, maybe some Australian ones too. But this is a map here of all of the projects on iNaturalist that were had the word BioBlitz in their title. So as you can see, the vast majority are in North America, particularly the United States. There's quite a few in Europe. There's quite a few in Australia, at least the populated parts of Australia and New Zealand. But you can see there's quite a few from South America, particularly Argentina and Chile. iNaturalist is a multilingual platform. It has a multilingual app. The Spanish version is quite mature now, and so it is very accessible to other languages. I think they cover something like 30 different languages, so it's quite good on that respect. Looking at Europe, though, Europe has a sort of older tradition on these kind of things. Italy doesn't have, I believe, any sort of central citizen science organization, and so you get a lot of Italians using iNaturalist. If you go to France, which has some very well-established old apps and organizations for citizen science, you don't get them on iNaturalist because they've got their own platforms, basically. And so you're still seeing a biased, so to speak, picture of where BioBlitzes might occur, but that gives you a general idea too. Just out of interest, maybe you could put your hands up. Who of you have all participated in a BioBlitz? You can give me a hands up from, can I do it from here? I need reactions. I see 5 of you, okay. And did you enjoy it? Okay, at least a couple of smiles. Good. So at least we have some experience in the room, and I'm guessing were those in North America or were those in Europe?
Europe,
Okay.
Portugal.
Good.
Europe.
Okay.
Also.
Perfect. So I can see there's some Portuguese dots on the map too. So I've mentioned iNaturalist several times. That's because we use iNaturalist quite a lot in our investigation. It's all, well, the people who set it up, I think in 2008, which is not that long ago, set it up with the focus on it being a social network for naturalists, not particularly for getting data for biodiversity, but obviously that's a very important and relevant application for it for us. But their primary aim is to link together amateur and professional naturalists and give them a forum, much like Facebook gives lots of people a different forum, to discuss, to identify, and to share information about biodiversity. And I think that's one of the beauties of iNaturalist, is that it allows people to manage their own data. You can give whatever license you want to your photographs and to your images and your sounds. You can decide what you want to do. You can put in your own projects. It's very much about letting the citizen decide what they want to do in terms of gathering information about nature, not about what the scientist wants them to do. That might be disappointing for those of you who were talking about bias earlier this morning, because it doesn't really help very much with that. But it does massively increase the engagement of people in it, because they're doing it for their own enjoyment. They don't feel they're being exploited by anybody and the data disappearing in a big black hole. They're actually doing something that they feel contributes to biodiversity knowledge, but they're getting a whole lot of pleasure out of it at the same time. So you have people on iNaturalist who try and make a record every single day. And I can't remember what the record is, but it's really ridiculous. It's like somebody has made a record on iNaturalist every day for the last 3 years or something like this. So you have a lot of catching up to do if you want to beat them. So, and you can see my profile here, so you can see everybody's profile on there. You can see I'm most interested in plants, and my favorite plant being Oxalis corniculata, which I'm sure most of you know and you probably have in your garden. That's that one there. And Bellis perennis, because before the pandemic, I used to travel a lot, and I always used to make iNaturalist records everywhere, and so I can actually go and see my map of where I've been, and so it gives me something back that way. Nothing to do with biodiversity at all. It's just fun to have these sort of memories of where you've been and what you've seen when you were there. Okay, that's iNaturalist. So I mentioned projects in iNaturalist before. We ran this one in Akrotiri, which is a peninsula in the southernmost part of Cyprus. I can show you a map in a second, but there was a few things I wanted to show you on this. This over here, where it says about, that's the description I made the word cloud of before. And so those are the words that people use to describe their bioblitz. Of course, that can be in different languages. It also gives you some hints here about the sorts of things we saw. So this particular little liverwort here was quite interesting and quite rarely recorded on iNaturalist. I also managed to find a rare lichen that was identified by our lichen expert at the Botanic Garden while we were there. But you can also see invasive species. You've got sounds that this is one Tim found, and Tim was actually I'm just trying to remember. So one person got the highest number of species, which wasn't Tim, I think. I think Tim got the highest number of records. So you'll correct me if I'm wrong. Yeah, he looks like he's still online. Oh yeah, thumbs up. So what you need to do apparently is record birds, because they're too easy. I was too busy looking at funguses and things like that. I put the title right over here, but I can go through quickly. We found 500 species in our project, so it was only 24 hours. A lot of experts who knew a lot about biodiversity, so that helped us. 1,900 observations. Only 52 observers. There were people from Alien CSI, plus we roped in some of the local people at the I think it's a conservation center from Akrotiri. But if you look under here, it says 236. That's the number of people who helped us with identification. So once our pictures were put on iNaturalist, people can come along and identify them, argue with us as what has happened. And every record on iNaturalist is always open for comments and re-identification, and that often happens. So I've had records that are 2 years old, and someone comes along and says, no, you're wrong, that's something else, and quite often they're right and I'm wrong. And so I should be much more cautious in my identifications, but it means that the data that's going to GBIF, for instance, can sometimes be in flux. So once it's got 2 good identifications ahead of everything, it goes into research grade, and those research grade records go onto GBIF. If somebody then comes along 2 years later and says, no, that's not whatever it is, then it can be changed back from research grade into just the non-research grade, whatever they call that, needs ID, I think. And so suddenly then the next update to GBIF, which happens very regularly, that becomes unresearch grade. And so you have to be aware that these data sets are not stable, even for things that have been identified quite a long time ago. That's a sort of throwback to what Lynn and Hanno were saying earlier on, that if you have repeatable workflows, for instance, use the iNaturalist API, and you're able to run them on multiple occasions, it doesn't really matter if the data set underneath is changing, because you can keep running it and you can see the progress in your data set. Obviously, if you want to publish that, you need to make a sort of a point in time and take a sort of data slice of when you actually process your data, but that can be done. But if you hadn't done it by creating scripts and by creating workflows, you will have, for instance, at the start of your PhD, downloaded all the data from GBIF, spent 3 years processing it, analyzing it, and everything like that, coming out with a lot of models and things at the end. And then you go back to GBIF and you find that half of the things you found you downloaded before will change their identifications, and you won't be able, you can't go back through all the spreadsheets you used to get to that endpoint to redo all your analysis. So making repeatable workflows, although it's quite a big hit early on, is actually a massive saving in work later on. So anyway, that's a lot of stuff I've said there. That's Akrotiri. We've talked about the bias before, and this is quite normal to have bias. I don't really even think of it as bias. It's just part of the method we used. And the BioBlitz method is not a good one if you want even spread of effort. So this is a big salt lake where you get flamingos and things like that. This is all a big biosphere reserve. It's very important for migrating birds as well as resident birds. And there's a lot of plants there as well, so something to keep me interested as well. There's a big Air Force base here, and above that grey line is Cyprus, and below that grey line is Akrotiri, which is one of the UK's overseas territories. Just to hang on from UK colonialism, I'm afraid. So the environmental centre where we spent our time was here, where it says 480 something. And as you can see, we obviously get the most records right next to the environmental centre where all the people are. And then during the BioBlitz, we went off in different teams, and you can see there was a team up here surveying this particular area, which is all salt marshes and reed beds and some dunes as well. But they have less records. there's still quite a few. Over here we have a lot of eucalyptus plantations and things like this, and you have fewer records because there's actually quite a fewer species there. But this is certainly not even. And I guarantee in this data set, there are a bunch of flamingo records, and the flamingos are somewhere in the middle of this salt lake. And you notice there's not a single record in the middle of this salt lake. I guess all the records of the flamingos are somewhere in all of these, because the records of the flamingos are taken where the flamingo was observed from, not where the flamingo was. So if you want a map of flamingos, what you're going to find is they're not in the middle of the lake, they're actually all around the edges, which is a terrible bias, really. However, one of our topics we were looking into for bioblitzes was, do you get a decent inventory? And I think the results were, yes, you do. You will never get a complete inventory from a 24-hour bioblitz, but we were able to pick up a lot. So I filtered this by the birds and reptiles and amphibians, I think, because because always plants come to the top, and I know people are more interested in the sort of furry and moving things. And again, you see some of the biases. Flamingo comes out very high in that list. The francolins also, many of those francolins, most people didn't see. So a francolin sits in the middle of dense bush and sings, if you call it a sing, it's sort of a croak more. And so lots of people heard francolins. We also managed to catch francolins on camera traps, but there are very few pictures of francolins. And what else do you see there? Yeah, there's some amphibians and reptiles, because we had some herpetologists with us who must have turned over every single rock in Akrotiri in the process. So I would say bioblitzes certainly scores well for creating an inventory, but not necessarily a completely unbiased inventory. Obviously, more focused on these, one could say, charismatic creatures, the furry things, the feathered things. But we still picked up some other things as well. But I would say for the really rare stuff that we got, it was actually me who got the rarest one, I think. But it's because I was looking for really rare things. So I'm not interested in furred and feathered things. I look for all the things everyone else would miss. And so you have to crawl around on the ground and look on twigs and things like that. And so if you're running a bioblitz, invite people who do those weird and wonderful organismal groups. And even though I don't do those myself, I know people who do, because I work at a botanic garden. And so I also had a permit to collect things there. And so I was able to take things back and get them identified properly. The whole permit thing is a whole another question. Actually, that's good. Another question. How many of you have actually ever applied for a permit to go collecting anywhere? Good, at least one person. Was it easy, Joachim? Oh, we can't hear you.
Yeah, I applied for a permit to catch podarces in Portugal, and it was really easy. We got it in about a week or something.
Is that an invasive species?
No, it's natural species, native.
But not a rare thing then?
No, it's not rare at all.
Okay. I think
but if you are from a research center or legal institution of biodiversity conservation will give you a permit without any problem, rare or invasive species. If it is for scientific purposes, you get it easily.
Okay, that's good to know. I should come to Portugal. Yeah, so I've had very mixed. Some places I've had very good experience, and other places it's been extremely difficult. But I always think it's a bit difficult if you're going to another country as well, where you don't know the political situation and things like that, and you don't know the language, then it's very hard to discuss what is or isn't allowed. But I do make a point of making sure I do get permits for things. I know certainly within Europe, it's very tempting when you're on holiday to collect things, but the way the political situation is with the Nagoya Protocol and well, just for the respect to the other people's countries, that getting people's permission to collect is a very important thing these days. Right, inventory. Early warning, yes. So we particularly focus on invasive species and early warning of invasive species. Now, there's some things about early warning is if you're running a BioBlitz in a particularly in a nature reserve or something like this, you're not actually likely to pick up first records of alien species coming into a country, which may come into gardens or ports or and trade. You will get maybe early warning of things establishing in the nature reserve, but nature reserves are generally not the first place invasive species come in. So I wouldn't say a BioBlitz is perfect for finding new species to the country. Having said that, we were still able to find new records for invasive species, or at least alien species for Cyprus that had not been, I wouldn't say they'd not been seen on Cyprus before, they'd not been recorded in the wild in Cyprus before. So I'm particularly thinking of a cactus. We found one native species that we think was new to Cyprus, but it was a shell. And so we're not really sure where that shell may have come from. And so, I mean, it's another thing we were talking about photos before, and I think having photos of all of this stuff is very, very useful. Again, you can see from these pictures here the bias of people's records. So Oxalis pes capri was absolutely everywhere on Akrotiri. We also did this survey, I think, in end of February, beginning of March, and that's exactly the time that Oxalis pes capri was flowering. If you went back there in August, you wouldn't even see any Oxalis pes capri. All the leaves would have died down and everything. Also, charismatic plants like cactus tend to get picked up quite easily. I think next to the Akrotiri Nature Center, there was like 1 or 2 plants of Opuntia ficus indica, and everybody made an observation of Opuntia ficus indica. It's a bit like, so I did a paper not so recently where I was looking at captive and domesticated organisms, and I think there are 25 billion chickens in the world, apparently. That's a lot of chickens. But if you look, oh no, I've got that wrong. No, maybe there's something like 4 billion chickens. It's a hell of a lot of chickens in the world. But if you look in GBIF, how many chickens there are, there's 25,000 records of chickens, which is not many records of chickens. Now, you might think, well, why do you want records of chickens on GBIF? And I was comparing them with the bearded vulture, which has 24,000 records on GBIF, which is probably about a quarter of the world's population. And probably the same bird has been photographed multiple times. Certainly the Californian condor, the records on GBIF, there's way more records on GBIF than there are Californian condors in the world. So you really have to know your organism and the rate people take photographs of it to understand it. Getting back to, I shouldn't really go on about chickens, but the chickens are very important for things like transmitting bird flu, and chickens are not separate from the rest of the world. They are part of the ecosystem. In many parts of the world, they mix very well in the ecosystem, and they carry a lot of diseases between human beings and wild animals. So chickens are important, as are other domesticated animals. So BioBlitzes, yes, good for early warning to some extent, but maybe not perfect. Engagement. So this is a bit hard to explain. By the way, does anyone have any questions up to now? Oh, go ahead.
Sorry, I'm assuming the BioBlitzes are mainly for terrestrial surveys. Am I wrong?
Not exclusively, but I think you're right. I think we actually tried to have a marine aspect to the Akrotiri one. Someone bought an underwater drone. I don't think we managed to get it did work, and they scooted it around. We don't think we got a single observation from them, though, because it was a bit hard to identify anything. And the local scuba club were going to come along, but Akrotiri is a very difficult place to dive, and the weather forecast was bad. And so I think for lots of reasons, like if you have a set day and the weather's bad for scuba diving, your marine BioBlitz is off. It's not a good format, I think, for marine. But it's a good question.
Thank you. Yeah.
Or I guess if you wanted to do shoreline surveys, then you need to make sure it's low tide during a BioBlitz. Vanessa, did you have a question? Oh no, okay. So let me talk about engagement. So I'm going on about iNaturalist, but it really does have a fantastic wealth of information buried in all the data there. And you have people like myself who have got recording histories over a long period of time, and we have our user ID. And what I was able to do was I randomly selected 100 BioBlitzes that have gone on as projects onto iNaturalist over the past few years. I then extracted all of the users that participated in those 100 BioBlitzes, which was about 33,000 users. And I took all of the records that they made for the year before the BioBlitz and all the records that they made for the year after the BioBlitz, and I subtracted one from the other. In actual fact, I did it, the numbers of days they worked, they did a record per week. So because obviously for seasonal differences, organisms are more or less accessible to record in the winter and things like this. This is why I did it, the number of the activity per week on iNaturalist. So if I've explained that right, you'll understand that if in the year following the BioBlitz, they were very active, they could do a maximum of 7 days more activity than they did in the year before. Or if they were less active, they could do a minimum of -7 days activity compared to the year before. So that's what you see on this graph here. On the right-hand side, you see the full graph. So it's jittered, so you can, because it goes 1, 2, 0, 1, 2, 3, 4, 5, 6, 7, and that minus. So it's jittered, so you can see all the points. And then we fitted a logarithmic decay to this as a model to fit it. So we fitted a logarithmic decay because it allowed us to calculate the half-life for the activity. So the BioBlitz is at time 0 here, and the boost is anything above the line, and the half-life of their engagement after the BioBlitz is then shown, and the half-life was something like 12 weeks. So as it happens, my wife works in marketing, and this is a good result. So if you run an advertising campaign, getting people still to be buying your product 12 weeks after your last advertisement is a good result. It also means we were able to calculate how many more records that we got compared to if we hadn't run the BioBlitz. And I possibly need to change. I need to go look at the paper because I can't remember the details now. I probably lost it. Okay, I can't remember exactly. I think you'll have to believe me that I think what we got was, yeah, that's right. So for every person that took part in a BioBlitz, they did 7 more days activity in the year after. Now 7 doesn't sound very much, so over a whole year 7 is not that much, but it's 7 more than they would have done anyway. But if you've got 33,000 people participating, in a BioBlitz, that 7 adds up to an enormous number of observations in a year. So what I can't promise is if you run a BioBlitz in your nature reserve, that the people who came to the BioBlitz will be, that engagement will make them come back to your BioBlitz, your nature reserve, and do all observations there. What probably happened is they came to your BioBlitz and they do new observations elsewhere, which is great for everybody else and potentially for you, but it doesn't mean they're necessarily coming back to your nature reserve to give you more data. So I think the take-home message in here is that, yes, you get a really big hit with engagement from running a BioBlitz, and you get many more records, but not necessarily the records you want. Okay. Any questions on that? Did I explain it all? I know it's a bit. Okay. So we're back onto learning. Now, learning didn't come out massively high in our literature review. By the way, this is from the Akrotiri BioBlitz. The reason we're wearing funny hats is because it's carnival time in Cyprus. And so this is me, I'm a flamingo. There's a couple of flamingos, and this is Tim, I think, here with his punk haircut. So we're learning about this. We're learning about the marine drone for looking underwater from our representative from Malta, whose name escapes me at the moment. Anyway, so a learning aspect is not absolutely essential for BioBlitzes, but the literature seems to show that if you put learning into your BioBlitz, it means that people come again next year, and they'll also have a much higher response to your BioBlitz. So the fact people learn new things makes them feel more engaged. So a lot of that hit we get from engagement is actually coming from learning. So even though we listed learning and engagement as separate things, they are rather intertwined. Well, I don't have any data on learning. It's a difficult thing to test. You have, if you're going to run a BioBlitz and you want to know about learning outcomes, you really should do some testing before and testing afterwards and try and figure it out from that. And we didn't do that. I'm guessing none of you have actually run a BioBlitz, but for those few people who had been to a BioBlitz, did you feel like you learned something? Even if it was just identifying one species?
I went to a BioBlitz, but as a monitor, so so I expert on herbs, so I don't have the perspective of the participants.
Well, kind of the experts still, they're still participants, but it is a different experience for sure. And were you, when you were working as an expert, were they just bringing you plants to identify, or did you actually sort of show the people how to identify them?
I'm not a plant expert, so I focus my research or looking for some herbs. That is my research expertise.
Oh, you mean like herpetology herbs? Ah, okay, sorry.
Yeah,
yeah. Yeah. So okay, so they didn't bring you lizards and snakes, obviously, but maybe pictures though.
Yeah.
Yeah. So I don't have much more to say on learning, only that I believe it is important, and it's important to take into consideration for a BioBlitz if you're doing it. So if you remember right at the beginning, promotion and organization came least on everybody's literature. I don't believe that's really true. I think people just don't want to say that they were trying to promote their own organization. I think promotion of an organization is important. I think in many Western cultures, we just don't like to be self-promoting so much. And particularly in science, it's kind of frowned upon to be pushing yourself too much. However, if you look at all of the BioBlitzes we had in the literature, I guarantee that all of them had participants from different professional organizations involved in biodiversity. So they may be conservation organizations, they may be museums and herbaria and botanic gardens, but all sorts of experts. And I think one of the big things about BioBlitz is that the networking amongst professionals is just as important within the BioBlitz as it is about networking with citizen scientists and expert citizen scientists as well. So I put a picture of a network here because I think a BioBlitz is a way that basically biodiversity nerds can find each other. Normally they're buried in the jungle somewhere looking for interesting organisms, but a BioBlitz allows people to get together and talk about things of mutual interest. Maybe they don't get a normal chance like me. Unfortunately, my family are not remotely interested in biodiversity, and so I can actually find someone to talk to about this kind of stuff. I'm looking around. I want to hear what your feelings are about this as well. Do you think I just need to find new friends?
Quentin, can I say something?
Of course, yes.
Hi, so I actually also promoted BioBlitzes, and I see that promotion it's really great to see other people to talk about, but also to bring these kind of ideas and these kind of discussions to people that normally don't do it. So I actually like to bring my family as well. Normally they don't like biodiversity also, but for example, the last ones that we organized, we had a concert in the middle of the day. So we brought people for the concert, and then they stay for the rest of the day. We got a little trigger to talk about biodiversity, and now the next time they want to come as well. It was about trees and the biodiversity that lives in trees. And actually, one of my family members just bought a little, what's the name? The little glass to see lichens and bryophytes. So they just bought it. So when they go for a hike or something like that, they go to the trees and they look at themselves. So it actually has this kind of things. So it's not to look for the other nerds like ourselves, but to bring a little bit of this to common people, if we can say that. So yes, it's great. And yeah, just share a little bit.
Yeah, thanks. That's really nice. Anyone else got any experiences? Okay. So I'm almost at the end, and then we'll go on to open refine. But so the recommendations that we're working on the paper, and it's not quite finished. We have next week to really hammer these out concretely. And probably there'll be additional recommendations out of this. But what we feel about BioBlitz is to make sure everything is very well documented. So we heard this morning about metadata. Most of these data on iNaturalist get put into projects, and when these data go into research grade, they go onto GBIF. But once they're on GBIF, they're completely separated from all their project data. It just goes into the iNaturalist single data set. So when people do modeling, like Bernd this morning, he thinks they are presence only data, and that they're ad hoc records, and he treats them like that. Whereas the people who collected it originally know how much effort was put into it and can potentially say, well, in actual fact, we can calculate absence records because we intensively surveyed this area. And so we're actually losing a lot of information in things like iNaturalist. And I moan at iNaturalist all the time, don't worry. But this loss of metadata is quite important. So you can publish your own BioBlitz data outside iNaturalist and things like this, but of course, things like iNaturalist make it too super easy to collect. So good documentation, documenting the aims, documenting the participants, obviously with their permission to use the data and things like this, and to give credit to them. the results you get and the interesting things you find, and what you were trying to do in terms of learning outcomes potentially. I'm always banging on about open data publication of data, because I know if you keep it on your computer, after a few years it will have just disappeared and you won't be able to understand it. And so if you put it out there, you will make sure it's good enough to understand and people can still reuse it and understand it. Not writing your own app, but using somebody else's app is super important. It used to be more common that people would do this. Now people are realizing that they just can't compete with some of the bigger apps, and they just do their job too well. And leverage it, so you're effectively leveraging the things that are out, the tools that are out there for you to use. Verification of records is super important. There are many discussions. I could probably find some from our Acritary BioBlitz, where we got massive great discussions on the identity of particular taxa. Getting professionals involved is a very common theme in BioBlitzes. Getting locals involved is super important to get those sort of goals in terms of engagement and learning. Keeping it simple, but as you saw in Acritary, we had a very simple methodology. Basically, you go wherever you want and you record whatever you want. It doesn't produce very reproducible results, and so that's something we need to think about. And inclusivity is, I think, certainly in Belgium and in the UK, which I know, it tends to be middle-class white people who are involved in BioBlitzes and not inner city children from ethnic minorities. And they have probably got the benefit and the most learning they could do from that kind of thing. And we probably are taking the easy route by running BioBlitzes in nice nature conservation areas rather than going to an inner city park and running a BioBlitz. But one should consider the value of doing that. And so there's lots of different ways you can engage for inclusivity, including things like prizes and games and locating your BioBlitz in different things. So has anyone got any recommendations, any comments on those recommendations?
Yes, I want to share an experience. I think it's regard to use this data for species distribution modeling. Once I looking for Icornia crassipes, water jacint here in Sardinia, and I saw a record, and it was a little bit strange for me because I know where is this species in Sardinia. So I go through this record and I call this person and I say, okay, you put this record on in Naturalis, and she said, yeah, because I saw this species in like artificial pond inside of the hotel. And I say, oh, okay, I think it's not very it's not accurate, this kind of data, but for the rest I think it's a great tool. Actually, once I opened a project for Ailanthus altissima, and there's a lot, a huge quantity, and this was a kind of competition between in our department and with some colleagues. And when I see, okay, these people collect data, and I go through for more data and so on. And this is a good motivation to collect a lot of data for a particular species. So it's like a funny way to collect data. It's a good experience. Nice. Yeah.
Do you want to say something? Nadia?
Yeah, no, I have a question. I was wondering how the workflow is between a naturalist and your BioBlitz. So with the upload of photos, the identifying of species, so is everyone uploading photos? You know what I mean?
Yeah, so we encouraged people to use iNaturalist, so they did it on their phones. Some people took their cameras and took photographs and then uploaded them that way. And there were a few people who gave me lists on pieces of paper, and I had to then make them into Darwin Core and publish them onto GBIF independently, which is a heck of a lot more work than putting them on iNaturalist, but I did it anyway.
So there is more or less no workflow, but everyone's doing as they like, more or less.
Well, I mean, there's a lot going on behind the scenes, I guess. We also, for the few organizers, we made a point of going in and doing identifications on the record, so that for the project-based observations, we'd identify as many as we could. So in terms of work, is that what you meant by workflow?
Yeah, so for example, for persons who have no identification skills at all, and there is no expert, what do? They just go around and take photos?
Yeah, they
see?
can put unidentified things on iNaturalist, and very quickly people come along and put an identification on them.
Okay.
So, well, of course, the iNaturalist has a machine learning algorithm as well that'll help identify things. And it works reasonably well. I mean, it works very well for me, because normally I know what the species is, but it'll be in the sort of list of top 10, and I can just pick the one it is. If you pick the top one every time, then you're going to get wrong quite often. But,
because as a participant, if you don't, so you would, yeah, the learning experience is not so great. So you would recommend to have like many experts, like
yeah, I think
one expert for 5 people more or less to.
So what we did is we made up teams, mixed teams, where we were trying to balance experts in different organismal groups with also local people. So we would try and sort of put a bird, a butterfly, and a plant person in the same group with a bunch of people from the locality, so that but it's impossible to even it out totally, and that was, it was also good to get people away from the environmental center into different areas of the area, so we're not all just identifying the same cactus like we were around the environmental center, for instance.
Okay.
But I think we could do a lot more work in improving the methodology, and that's if we get invited back to Cyprus, that's what I plan to do. So I've managed to rabbit on for an hour. I hope you found that stimulating. I don't know, but we will now move on to the coding club side of things, and I'll introduce you to Peter Herbrechts, who is in my team at Miser Botanic Garden, and he's going to introduce us to OpenRefine. I think there's a few other people joined us. Those people are from the Alien CSI Coding Club. If any of you want to join the coding club, you're very welcome. We meet at this time every week on a Friday, and we will through till October, which is what I promised. And then I'm probably going to take a rest unless someone else takes it up. So Peter, it's over to you, I think.
Hi. So I'll be talking about OpenRefine. I am not prepared for like a real traditional style lecture, so I'd appreciate it if you have a question or remark, or if anything is unclear, that you just interrupt me. And I really mean like interrupt me. So so we can have some sort of interactivity here. It's all not very formal, this coding club stuff. Can I share my screen, Quentin?
You should be able to, I think. Maybe not. Sophie, is that, yeah.
Can you see my screen now?
Yeah.
Okay, so this is OpenRefine. It's running in Firefox on my computer. It's a Java program, so you launch it and you can view it in the browser. And so I'm just assuming that nobody's ever heard of OpenRefine here. It's a program that you can use to explore data. It's used for data cleaning. It's used for data reconciliation, which is the process of trying to disambiguate different values in a field. Then just tell me if that's complete Chinese to you. And it was first developed by a company that I'm forgetting. Then it was bought by Google, and now it's been handed off to the open source community. All right, about OpenRefine. OpenRefine looks a bit like a spreadsheet. but it isn't. It's actually a database running in the background. I think I can just better show you by example. I'll open just some files that I prepared in advance. This is a set of iNaturalist observations of oxalis by popular Quentin request. And this is a file from the Do That crowdsourcing platform that I'm also administering. So we can see some stuff that was entered by citizen scientists, because that's very messy and that sometimes needs cleaning. So let's start with the iNaturalist stuff to stay in the team. So this is all running on my local computer, just to be clear. So this is not running in the cloud. I think it's possible in principle, but not very regular. I have an instance running on our development machine in Mesa, but this is just my computer. So it's read this CSV file, and it's giving me the first few rows. And now I have lots of different options for the data import. It's correctly identified almost everything, I think. It is UTF-8, it is comma separated. I'm allowing it to trim white space, which means that if there's a space at the end of a cell, it'll get rid of it. Yes, it's got a header, and this is the quoting character. I can tell it, just like Excel, to interpret my data. So like if it thinks something is a date or a number, it'll automatically make it a date or a number. But I'm not going to do that for now for demonstration purposes. OpenRefine works in projects. Not in files, which may sound a bit confusing, but I'll try to clear that up. So you can give your project a name. I'll call this coding club iNat, and you can give it tags. So I think I've got a coding club tag, and I think I also got an iNaturalist tag. And then I'm say create projects. And now what it's done in the background, it's created a zip file with all my project data in it. It's not changed the original file. The original file is still there, and whatever I do in OpenRefine, it will not change the original file in any way. It will autosave, but if you want to get a CSV file or an Excel file or whatever out of it, you will have to click export right on top here. So this is OpenRefine. It looks like an Excel sheet. It tells me I've got 8,000 rows, but I can't scroll because you're only always looking at that many rows. I usually set it to the max, but depending on how big your rows are, you might want to set that a bit lower. What I usually do at first is just have a look at all my data. And something that's very useful in OpenRefine, and what I use it for mostly, is data exploration. So you could, if you've got like a really large file, have a look at what's in it by writing lots of filters in R or in Pandas or whatever you like to use, SQL, because you can also load databases and stuff into OpenRefine. I can actually show you that. So you can connect to databases or Google data, or even scrape directly from websites in OpenRefine. So OpenRefine works by rows. So every row is a record, and every column you can see as like a classical Excel column, but you can also imagine them as tables in and of themselves. So you could have like a complete parsed JSON file in a single cell if you wanted to. Something else that I should mention, OpenRefine is a very powerful program, and it has a lot of options. I don't know all of them. So don't get stuck in that. Just use it for what you want to use, and Google a lot. As you can tell, I've got a bunch of tabs open here in case of questions, and I'll probably use some of them during my explanation. But about facets, facets are like filters. So let's see what would be a useful facet on here. I'm going to be moving the little, my own face is in the way. Here, what would be a useful facet? So here's the species name. This should all be the same. So I'm just going to hide this column, collapse it. Species ID should all be the same. So collapse it, because I took this export from iNaturalist, and that's what I filtered on. So we don't need to worry about those. Oh, we've got here captive or cultivated. Let's make a facet based on that. Just a text facet will do. So now I'm getting all the options in there. They're all false. This should not come as a surprise because I set it to research quality. So everything that's captive is not allowed in here. Yeah, I'm thinking I should have maybe started with something else. Oh, here, license. So if I take a text facet here, I get all the different values that are in here, and I can filter on these values. I can also have a count on them. And for example, if I'm only interested in CC0 records, now I've got all the CC0 records selected, and every change I do from now on will only be on these rows. So if I add another facet, I will only get CC0 records. So let's transform this. So if I click on this little arrow here, I'll be working with the entire column. I'm just trying to demonstrate the power of OpenRefine. Please don't see this as like a tutorial because I'm probably going a bit fast for that. Let's edit the cells and transform them to a date, because I think iNaturalist is clean enough that I'll trust it to do that. But if I go now back to all my records, you'll see that I've only transformed the dates of the CC0 records. So that's something to keep in mind. So if I go back to CC0, now I've got the dates. And if I add timeline facets, I can actually filter on the date fields. So now that we've seen this, I'm going to move over to a different data set that's a bit less clean, because of course iNaturalist does a lot of the parsing in on the platform itself, and the data you get from there will probably be a lot cleaner than if you got it straight from a GBIF dump.
Peter, can you just do the timeline facet again, just very slowly, so people can see? I missed it.
Oh, sorry. So I'll do it for the complete set this time. So if you've got a date, you can, and it's very consistent like this one or this one, you can just do an automatic transform to a date. So what I'm doing here is I'm changing the type of this column. So at the moment, all my columns are strings because I asked in the beginning to not assume anything. And now I'm telling it this column is a date. Now it's transformed them into a date. Now I'm clicking here, I'm adding a facet, a timeline facet. And here in this timeline, I can change the width of my window and I can move it. And if I had really strange things going on in this column, it would show up here. It would say error or invalid. Or here, for example, with my facet I have on license, I have 420 records that apparently don't have a license at all.
Yeah, you can actually see the seasonal phenology, can't you? Because you've basically got the flowering period of size.
Yeah.
That's kept through there.
And something else that's interesting in the same vein, I've got these two strings here. So let me just these are latitude and longitude decimals. Let me just transform those into integers, which OpenRefine calls numbers to number. Although I guess they're reals. Yeah. Anyway, to numbers. There we go. And if I add a facet, I can do a scatter plot facet. Or at least I could. I'm not very good at getting the scatter plot facet to work most of the time. I'm not actually sure why.
There's lots of data.
Yeah, it might be that it's just too much. So that's also something to keep in mind in OpenRefine. While you have the power to do a manipulation on literally 1 million rows at a time, and it will let you, and it will be relatively quick, it doesn't mean like the more complex stuff will still work if you try to do it in 1 million rows, especially reconciliation, because what you're usually doing is you're asking Wikidata, for example, do you know what this plant is? And if you do that 1 million times, you will have to wait a long time. No, still doesn't like it. So I did try this earlier, and I also had some trouble, but I got it to work. I don't know why it's not working now. Shame. So what you would get is a scatter plot of locations, and if you do that with location, you can see geographical outliers. So you would see usually in the middle of your scatter plot at 0, 0, you'll usually get a big blump, which is people entering 0, 0 for coordinates manually. But then again, those might not show up because these are all research quality.
Could it be because they were negative in that column?
I don't think so. I did try this one as well.
Oh, did you? Okay.
No, I really have no idea why it's not liking this.
Could it be, do you need to perhaps select 2 columns?
Yeah, usually what it'll do is it'll give me all the in all the columns that I have the type number for, and I'll do a multi-axle scatter plot, and then you just select the one you want. So maybe if I set some more to numbers, that'll help. No, I have no idea. But I've had trouble with this before, and I think in this version, which is the latest stable release, the reconciliation engine for Wikidata is also broken again. So I got that to work, but I had to go to a different Sparkle endpoint, which is a very technical way of telling you that OpenRefine is great, but sometimes it requires a bit of Googling. Anyway, something else you can do is splitting columns. For example, we've got here time observed and the date together. You can do this sort of data wrangling exercise where you want to split a column. If you can find it here, split into several columns by a separator, and I think that should be a space here. Or you can use a regular expression as a separator. See, now it split them for me, and then you probably want to rename this into something else. This maybe to date observed. And this is the sort of thing you can do with OpenRefine. OpenRefine also supports a lot more powerful features. For example, if I make add a column based on this column, here at language, I have the option between GREL, which is an OpenRefine specific language, I think developed at Google. It looks a little bit like Python and regex had a baby. It's a bit strange. Then Python, which is actually Jython because it's a Java implementation of Python, and Clojure. I'm not really fluent enough in Python or Clojure to do a presentation here on them. So I'll probably stick to GREL most of the time. So what I could do this are just strings. Say if I want to transform them to dates, I can do that like this. There you go. So now they are date entries. And if I say I want to extract from this date a string and I want specifically the day of the string, there it is. And I can call this day, for example. And if there is an error, I want to set it to blank. If you're noticing you have a lot of errors, you may want to have a look at what the error is. So now I've created a day column, and now I can make, I can transform this to a numeric first. Common transforms, and now the faces are in the way again. To a number. There we go. And then I can have a numeric facet. There we go. And we can see it's mostly spread equally. So the Oxalis Pescapria is actually pretty clean. If you would do this with our collection management system, you would see two big peaks, one at the 1st of the month and one at the 15th, because those are used to denote a degree of uncertainty. And I think I wrote some GREL at some point. This is GitHub gists where I stored small pieces of code.
Peter, just to let everyone know, I put a link in the chat to the user manual for GREL as well, because there's really a lot of functions that people can use, and just looking through the user manual will give you some idea of the sorts of things.
Yeah. So just for an example, what sort of thing you could do with GREL is I wrote a facet for myself to select dates that had the 1st or the 15th as the start, as the day of the date, because those mean something to me in this specific case. So what I do is, in the case that one or the following values take the date in this format, select the date, and if it's this equal to 1 or to 15, give me true. And if it's not, give me false. So that's the sort of thing you could do with GREL, and you can really go quite far in GREL. I don't know what else I've written. Like, for example, if you've done reconciliation, we'll get back to that. Give me all the possible things that it could be on Wikidata or GBIF or whatever, and put them in a single cell, delimited by comma. Delimited by whatever you want. By a semicolon, there you go. I'm gonna, oh, this is maybe probably a good point. OpenRefine at this point has stored everything that I've done as a sort of sequence of commands. And I can go back So I've done a text transform here. I've done a text transform here. Say I want to go back all the way to the beginning. I click here, and now I'm back at that state of my project. I click here again, I'm back at that state of my project. If I click here and I make a change, the tree doesn't split. I just remove everything that I did before. So, and this you can also extract. You'll get a really long JSON string. You can also remove steps you've done. And the handy part of this that I've never used is that in principle, you could do a number of operations, then store them, get a completely different input data set, just load just paste the JSON in here. For example, I think I actually added, so if I would paste this JSON in here, it would just take that command and use it, but it won't work because I don't have the right columns. And if you take a bigger project, I'll show you one that I've actually done work on. So open projects. So for example, this one, which is from our collection management system. That might have been a mistake because it is really big. Yeah, that's a mistake. I'll take a smaller one for demonstration purposes. Then you'll see that that list can get pretty long pretty quickly. And that's what I think the power of OpenRefine is really big, and you can make repeatable science with it. But I think if you're really doing a very complex manipulation, you're probably better off writing a true script for it. But of course, it's all up to you. And I guess it is truly repeatable in OpenRefine as well. So let me just, this one maybe. Yeah. And if I hit export here, I can write the whole thing as an OpenRefine project. I can also export it as a comma separated file. And it's only at this point when I export it, I will actually get a file with my changes in. It's not lost to the ether if I close my browser now because OpenRefine has changed it, but there is no file I can browse to with Excel to open all these changes. So if you're doing lots of stuff, it's probably wise to make manual exports from time to time. I've learned this from experience. While we're waiting for this other project to open, this might actually be failing because the original file is just gone. Oh no. No, there it is. So here I've got a history of 671 steps. And you see that I'm here matching, because that's the title of the project as well. I'm matching strings to VF IDs, which should be in here somewhere, I suppose. Or maybe I'm faceted on the ones that I haven't done.
You'll have to explain what the VF ID is.
Yeah, so VF is an identifier for persons. Basically, everybody who's ever published something runs the risk of getting cataloged by a library. And all these libraries have come together, and to make it easy to tell Quentin Groom from all the other Quentin Grooms in the world apart, they have assigned them a unique number, and these are published on their own systems, but also very often on VF. And that's like very famous people and also very unfamous people. So I'm not even famous enough to have a VF ID, but I'm pretty sure like some people who are in the room will have one. You're my victim of the day, Sophie. Nope. But Quentin is. So VF knows that Quentin is a botanist. And he's got two IDs, one in ISNI and one in Wikidata. So by linking to VF, for example, I can get a Wikidata ID, and by going to Wikidata, I can get lots of other interesting stuff from about Quentin. So for example, his Twitter handle, his eye color is in here somewhere, I think. Isn't it, Quentin?
Yeah, I'm a bit out
yeah.
there. Really,
yeah, there you go. Your eye color is in there. Where you work, what you like to do, and here's all the other IDs that he has. So I could make a jump from a string of his name to VF ID to like his BHL creator ID. No, just one ID, his IPNI author ID, and so forth. But maybe it's probably better if I just show you an example like that. So I'll go to my Do That project. Dodat is the crowdsourcing platform of the garden. So we use it for taking herbarium sheets, which is a piece of paper with a plant stuck on it, and a label, hopefully. And we'll take the information that's on that label, and people will enter it for us. And then through magic in the middle, it goes into our collection management system. And then magic happens again, and then it goes on to GBIF. So you can give it a sensible name, but that's not. Store blank rows, create a project. So this is like a production level file, but one that has been anonymized. So I technically chose the one that's anonymized, so you don't see the names of our validators or transcribers. I'll collapse this column because it's a long string and we don't need it. And we can go to the back, and here you'll see a verbatim locality. So this is a column of names that people literally entered, and it's a lot more messy than something that you get that's already cleaned. But because the file is a bit big, and because I suspect that like when you're trying to do a cleaning exercise, you always should try to make it easy for yourself. So what I suspect when it goes to people collecting in the field, that one person is more likely to see more things at the same place over time. So I would do this collector by collector and going from the most prominent collector to the least prominent. So let's take a filter on our collectors and see who is the most prominent. I'll tell you right now, it's Philippe Verloven. So I'll filter by count. There it is, it's Philippe Verloven. And SC is scenic collector, the ones where we don't know the collector. There we go. So now I've only got 262 matching rows. And these are all the locations that the people think that he has collected. So I can make a text facet. There we go. I can make this smaller, but I'm leaving it on. For example, if I click invert here, I'll get everybody but Philippe Verloven. And if I click reset, I'll just get everybody again. There we go. So now it's still, like if I say everybody, for example, it'll tell me 287 choices total, too many to display. It's still got them, but it's just too many for it. And then here we go. So he's got collected in this project on 210 sets. And just looking through them, we might be able to find some manually that probably should be the same one. But because manual work is for manual people, we can try to cluster them. And this is where the very interesting math of OpenRefine comes in. So you can do clustering and all sorts of things in R and so forth, but OpenRefine really makes it very easy. And this is very good for data exploration. For example, maybe there is a typo in my data set of 8,000 rows somewhere. Using clustering, you can find them. So we're trying to find values that are subtly different, but probably are meant to be the same. And OpenRefine uses two methods to do this. So we're doing fuzzy matching. For those following along. Key collision and nearest neighbor. Key collision algorithms work by taking a string, so a number of letters or numbers, trying to reduce them to something that contains all the information, and then looking at all the other entries that were reduced to the same thing. And the thing you reduce to is called a key, and all the ones that are same are put in the same bucket or in the same bin. So you have different methods of doing this. Fingerprinting is you'll get rid of everything that people are likely to do wrong. So for example, everything that's capital letters just gets bunked down to non-capitals. All the commas and points are removed. Everything that's like emphasis is removed. All these strange encoding things are removed. Spaces are removed and the letters are reshuffled. So what you're basically doing is you're trying to get rid of typos in a specific way. It's very similar to Ngram fingerprinting, but that uses more complicated math that I don't understand. You can Google Ngram if you're interested. If you figure it out, you can tell me. Metaphone, Cologne, Phonetic, Deutsch, Monokov, and Beider-Morse are all phonetic matching systems, which is basically your computer using fancy math, trying to pronounce the word that's there. And things that sound similar are probably the same thing. Is the reasoning works really well for names, because often people will hear a name, they will write it down, but they don't actually know what it's spelled. Metaphone 3 is Soundex based, works really good for English. Cologne, Phonetic, you can hear it by the name, is German based. That's probably one you would use for Dutch. as well. Deutsch Monoktov is for Slavic languages, works really well for Yiddish names, and Beider Morse is another one that's adapted. I think that one's for Eastern European names as well. But something to be really wary of for everything that's fuzzy is that you need to know what you're doing. So you need to think about the clusters you're making. For example, if you see, for example, there's a classic example of a hotel that is the same name in London and in New York. Hotel Savoy and Savoy Hotel or something like that. If you just merge those blindly, you'll be merging two sites that are different just because they have the same name. So you do need to still think about it. So if we say Cologne phonetic, everything that sounds really similar will get merged together. So now it's up to us to find the difference. Huh?
Just wanted to check that everyone's following along and there wasn't any questions at this point.
Yeah, sorry, am I going way too quickly?
You're going quite quickly.
Sorry.
But is everyone following what Peter's trying to do here?
We are.
Yeah, you're right. I give you credit for obviously. Good. Okay.
So there are two rows that have this value, one with this, and one with this. And using this algorithm, I am investigating the hypothesis that this is actually all the same thing. So the first step is always trying to find what is actually the difference. And I think in this case, it's probably just a space somewhere. Here they put a dash between North and Castile. And this one, I don't know. And you can hit browse the cluster and then you can have a look and so forth and so on. And you can do different ones in a row. And if you're happy, you can have another look at them. If you think they're all the same, this is the new value you'll be attaching it to. You can change it if you want, of course. And then you say merge selected and closed, and you're done, or merge selected and recluster, because often if you remove the obvious things that are the same, the algorithm will have an easier time finding the ones that it might have missed earlier. But this, with very many data cleaning things, this is really easy in the beginning, and it gets harder as you go on. So let's just merge selector and recluster because we're not being very serious about this. Also, the choice of clustering algorithm matters and is something worth considering. Most key collision methods work really well for spelling errors. Nearest neighbor works slightly different. You are taking something and doing some sort of calculation on it, and whatever is closest to that result is what gets binned. And you've got a number of algorithms here. Liebenstein is a very classical one. You take the distance between two strings, and if you find a difference, you give it penalty points. For example, if you've added a letter, that would be a point. If you've removed a letter, that would be a point. Maybe you've substituted one, you can give that a point. And depending on the values of the point, you can give it a different name. So there's like a bunch of different Liebenstein algorithms. If you want to know which one they've implemented here, you can probably look that up in the documentation. Honestly, for clustering, it doesn't really matter which algorithm you pick. And PPM, that's based on the Kolmogorov distance. And that's basically the reasoning that if you've got a really good compression algorithm, so if you would like to compress the text in the data sense of the word, like make a zip archive, you get a certain size. But if you were to add to that text something that's very similar in information content and then were to compress it, then they would have basically the same size again. So if A and B are the same, the compressed size of A and the compressed size of A and B are the same as well, because A and B are the same, and it just need to store that it's repeated. And the more different they are, the bigger the difference would be, and that's the reasoning. And then you can set a threshold for how different things can be. This works really well for larger blocks of text, because if things are out of order or there are like subtle differences, but for example, in OCR, like sometimes it misses a letter, that will be really hard to pick up for Liebenstein, but something like Kolmogorov distance-based would pick that up really well. So to summarize, OpenRefine can find errors or things that look to be the same, but aren't. for example, here, the harbor of Ghent, Roden Huizendok, the different spellings of dock, and here a point instead of a comma. That would have taken me a while to find, and it's really easy to look over wrongly. Of course, you can do it all manually if you want, but that's difficult. Here, the capitalization is different. This is a really good example as well. It's like, we would have probably found this regardless of which algorithm we chose. Anyway.
Hi, I have a question here. So when you do the merge for the selected features, will it merge their respective values, like values in the other cells?
No, so what it will do is it will take all these values in the column that we are clustering, verbatim locality, and replace them all with whatever I put here. So if I say Harbor of Ghent, Farmstraat E of Grovendonk, I say, I know it's just Farmstraat E, the Harbor of Ghent, I don't need it. And at Grovendonk, I don't need it either. I do this, it will replace all these values with this. And we can try that out. So it's done this. We'll close this. Now, by name, we'll have to go to the F, C, E, F. Here we go. And here they are. So they're all intact, except the thing that I changed is now Farmstraat E. And I'm still, just to keep this in mind, I'm only doing this for the records by Philippe Verloof. And now I've got this filter on, and whatever I do now only happens for these 6 records. So I want to reset this. Now I'm working again on all the records by Philippe Verloof. One final thing. Do you want me to tell you more about clustering, or should I move on? Because you can talk all day about clustering algorithms. By the signs, I'm probably assuming that I should move on. Do we want to know more about what you can do to clean data, or should I tell you a bit more about reconciliation now?
I think it's good to see the reconciliation.
Yeah, okay.
I mean, I think you should just explain what you mean by reconciliation first.
Okay, so reconciliation in the strict sense of the word means that I have values in my data set and somebody else has values in their data set, and we're going to try to figure out which values of mine correspond to the values of theirs. So that means that I could have species names like Poa palustris here, and GBIF, which is a large international database of occurrence and taxon data. They maybe use a number to identify this taxon, but I can still try to connect the two based on the information that they might have about my data or I might have on their data. In the context of OpenRefine, we are going to try to take the string, find some other database that knows something about the entity that the string represents, so about this taxon. And now that when I've made a connection like that, I can try to take data from their database, bring it in my data set and see, well, that could be endpoint if you're just interested in that, or maybe I can use that to see if my data set contains any errors. So for example, I could take all these scientific names, look at their range and see if my state province is in that range. But that's probably not suited for a demonstration. I'm not sure I'll manage that live, but we can definitely give it a go. So I've got 262 now. I think that's probably reasonable to try to reconcile. So you go to the column. So you've noticed I'm always working from columns down. You're not working on a single cell. Well, I suppose you could edit a single cell like this. But OpenRefine is really meant to be working in bulk. So you make a filter and then you change whatever the filter is covering. So I'm going to start reconciling. And now I've got a number of reconciliation services installed. You can add these by just a simple link. So if you have a link for one, and you can just Google for them, you can add it here and then you add it. But I'm going to pick Wikidata because it's by far the fastest.
Where do you find those links, Peter?
By Googling. Roderick Page has a good blog. Oh, wait. OpenRefine. I don't know, OpenRefine v, no, GBIF, BIF, Recon. iFile. There you go. So here's a bunch of these links. I'll put that in the chat. And you can also make your own API if you want to. I've lost the chat. How do you get from the presentation view to the chat counter?
Yeah, when your thing is up the top, you have to go.
Ah, yeah, I found it. Yeah. Okay. You can also do your own reconciliation API, but simpler is that there is. Where am I now? Oh, here I am. It's simpler to if you've got a CSV file that you have other information about these plants about. For example, you've got like a taxon table with with all the information about specific species, and then you've got an occurrence table with all the observations you've done. You could connect those through an extension that exists, and that's the reconcile by CSV open refine. I don't have it set up at the moment here. Reconcile CSV. That's a separate program you need to install. So, reconcile CSV. Yeah, there it is. And that's really useful if you're doing it for large sets, because it's less load on whoever you are pinging. So if you do it for 1 million species, you're literally asking the other server 1 million times. So that's not very nice. So maybe it's worth just downloading their whole taxon table and doing it locally. And it's a lot quicker. And you can do it on data you already have. So that's some extra application. I probably have some installation details. I didn't think of installing it in advance. Otherwise, I would have. Maybe if we have more time later, we will. So anyway. I'm using Wikidata. Does everybody know what Wikidata is? The coding club people should know.
Yeah, I think wouldn't be bad to just show the website a bit. The coding club people know Wikidata because we had a coding club on it. But
yeah, so this is Wikidata. If I add, what is Wikidata? Wikidata is the database backend of Wikipedia. So if I go to Wikipedia. No, that's Wikidata, sorry. And I go to an article. This is the species we were looking at earlier on the naturalist. You'll see that a bunch of information is here on the side. And this is not actually entered in manually by people, at least not most of the time.
Yeah, it's a bit variable on that, actually.
Yeah, people still do it manually, but they wouldn't need to see. They use a species box. And if you use a species box, it'll actually get all this information from Wikidata. But even if you don't, it's still connected to a Wikidata object. And this is how it knows where to get the images. This is how it knows what all these identifiers for this taxon are. All this stuff is stored in Wikidata, and you can go from Wikipedia to Wikidata by using this button here under tools. So if you go down, and this is for all languages Wikipedia, and all this information on Wikidata is regardless of language. So if you put something in Wikidata, it's in principle, you could show it on any language of Wikipedia. So if we go down all the way, we'll see that these are all the languages this Wikidata object is connected to. And it's also connected to Wikicommons and Wikispecies. I'll just scroll up again. And this tells us, so Wikidata works in triplets. It basically means that you've got an object, Oxalis piscapridae, with this number, has a property called is an instance of, and what is it? It's a taxon and it's an invasive species. And then it's got another property, it's got an image, and this is the image. And it's got a name, and this is the name. And this name has got an author, and that's the author. And this name was published in a year, and that's the year, and so on and so forth. And these are the identifiers we were talking about earlier. Oxalis won't have a VFID because it's never written a book, because it's a plant. Although you'd be surprised about what actually managed to get into into VF. Sometimes I wouldn't be surprised if somebody managed to get a plant in there. I know there was a cat that's actually got a VF ID, and I doubt that one wrote a book. Anyway, easy to get distracted. So for example, if we're interested about what we could get from Wikidata, is we could try to get a rank. We can have a look at common names. Maybe we are interested in getting the fact that it's invasive, that sort of thing. If you go for really big taxa, like a really well-described taxa, which is really well-described, Perea maybe, this one's got more information. Of course, you're dependent on what's in the database. And you're not limited to Wikidata, by the way. You can use your own CSV, your own database, and whatever else you want, GBIF, for example, as well. I'm just looking for a good example of what we would want to get. So let's say we are just interested in getting that taxon common name in English. Here we go. So we know we're only interested in taxa. For example, if you don't know, you can say reconcile against whatever type you want. If your taxon is your type you're looking for is not in this list, you can give it in yourself. But I know that everything in my scientific name column is a taxon. None of them are scholarly articles or watercolor paintings. And sometimes if you're having trouble matching, or if you want to do something specific like a person, because there's probably many people called Pieter Huybrechts or Sophie Meus or Heliana Texiera, then you might want to add other properties it could use to find the right one. For example, birth date would be a classic one. That's probably what I did here. So that's why you have all these birth date columns in this column, and like where they're from, and that's all the sort of stuff I would use to find a person. Sounds creepy, but it's very useful. Then I'll hit, I don't say auto match because I want to demonstrate that, but you could tell it like, if you're really sure that it's the right one, you can go ahead and assign the property. I'm not going to tell that. And you can also tell it how many do you want to see, because if it's uncertain, I'll give you several options. You can say, okay, I only want to see the first 5 ones. If you find more than 5, I'm no longer interested. I'll start reconciling here, and this will take a while because now it's going to ask it 262 times. And I said a while, but apparently it's quick. But if you do this for GBIF, it's actually pretty slow. So that's why I picked this one. If we go back to the article, he also, Rob, I think, made this. He also made it for the Encyclopedia of Life and CBI Worms, really good if you work on fish, and the Global Names Index. And of course, if you connect to Wikidata, for example, you can then take that information you got, like you know which Wikidata ID it's got, and you can use that to connect to other things again. So you can really hop around, and like even if you're only able to connect to one database, maybe you can use that database to connect to something else using the identifiers, for example. Okay, so Panicum capillare, species of plants. So if you're doing this for real, you probably want to be a bit more thorough. I'm just going to say that this is the right one. I'm going to tell it this record is this one, and everything that's got exactly the same string here is also this one. Like, for example, if you're doing this with people names, this wouldn't necessarily be true, because maybe you know here that this one is born in that year and this one is born in that year. And even though they've got the same name, they're different people. But for taxa, and especially for plants here, we're pretty sure that if it's got the same taxa name, it's the same thing. So I'll hit this button, and it's done 43 of them now. Pao palustris, it's 100% sure. I agree, Wikidata. Again, if you're doing this for real, please be more thorough. Then this one, okay, I'm pretty sure, and so on and so forth. And then once you've done a number of these, you can try to get data out of it. Also, you've got a number of tools that can help you doing this. For example, best candidate score. Maybe you only want to have a look at records at the beginning that have a really good score, like where it's really certain that it's found the right one. Here, for example, it's equally sure between these two, but this is the genus and this is the plant, so this is the correct one. But maybe you also want to have a look at the really bad ones, and then when you, like, for example, it doesn't know these ones. Oh, it's because it's got species in the end, and Wikidata doesn't use species in the end. So maybe now we want to use a transformation function here to get rid of this species. And you could do that with regex. So you would say replace a space followed by sp dot and followed by nothing else. Replace that with nothing, for example. And this is how you would find these things. But anyway, let's say that we're happy with the work we've done and we're completely finished, can't get any better. Maybe now we want to get some more data from these, and we go to reconcile, and we move the faces, and we say entity identifier. So no, that's not what I want. Match to each best scanner. So if I click this, it will just say match all the ones to the ones you think are best. So you might want to do that if you started from the, so if you've checked all the bad ones and removed those and all the rest is good, then you can click that button and you're done. Here, add columns and reconciled values. That's what I wanted. So here are all the properties it suggests to me. But these are not necessarily all the properties all these different Wikidata objects have. We were interested in taxon common name, weren't we? So here we go. And what do we see? These taxa actually have more than one taxon common name in different languages. And it's found all of them. And what I could do is I could say, oh, I'm only interested in a certain rank, or I only want the ones that have references. But let's say, no, I want all of them. And I click OK. What it's going to do, it's going to duplicate all my rows. So this all looks fine. But what has actually happened is instead of 260 rows, I've got 260 rows, each with a duplicate below it with the different languages. And if I click the records button here, it will switch view. And now it will put them here. But if you're, I can't really tell on my screen, so you probably can't tell either, but there's actually lines in between here. And then you can do stuff like fill down. And then you've got and then you can publish it as a CSV and you will get duplicate records, but everything is the same except the taxon common name, for example. And what you could do is you could get properties from this taxon common name if you are only interested in one language and so forth, although you could have done that earlier as well. But I just wanted to show.
Peter, can you select the language of the common name that you want?
Yeah, so you can either do that after this step. So now I can get properties of these names and then take the ones with the right language. But it's way more efficient in this case to just only export the English ones if you only want the English ones. But I did this on purpose so I could show you that OpenRefine, especially when you're working in reconciliation, you can really quickly add a lot more data and everything remains the same except the data that you're adding. And with this record view, it's easily visualized. But if you were to export this, like in a JSON, you can have this sort of thing because you've got a one-to-many relationship. If you want to export as a CSV, you probably want to fill down. And then whenever you want to do analysis, you would probably want to identify in some sort of unique value. On Wiki, do that, you would use task ID. If we go back to our iNaturalist dataset, I also got an ID. It's in general not a very bad idea to add an ID if you don't have one. We could do that pretty quickly, actually. So we could add a column based on this column, and instead of value, we could just add GUID. And I'm sure there is a way to add GUIDs in OpenRefine. We can just look that up really quickly. And this is really how I advise you learn OpenRefine. Just try to do stuff and then Google it. And if there isn't, maybe then we should, what's the alternative? It's not GUID, it's global, it's universal. So apparently somebody else managed to do it. Anyway, what I meant to say is that OpenRefine is in general very Google friendly. If it's not in the manual, you can probably find it on Stack Overflow. And Grell looks intimidating at first glance, but it's really not that bad. But here you can see as well, so I've got this value and then nothing for 6 rows and then this value again. That's because I just added all those vernacular names. And say if I want to do that again, so say if I want to add another property, it would do it again. So now I've got 5 times duplication. If I did it again, and then there's 3 times duplication, and I've got 15 times duplication and so forth. So that's why the records view is really handy. And if you don't want to deal with all that in your export, then you just export as something that is more suitable to that sort of thing. So to a JSON file.
Just to
Or you
give an
can
example of where reconciliation is really handy, if you have several data sets, you're all trying to get them all to the same taxonomy, you can go through all the data sets and try and harmonize them into one set of Latin names.
Yeah.
But a far more efficient way that can be more repeatable is to reconcile them with a single data set, like IPNI or with the Catalogue of Life or something like this, so that they're all reconciled with the identifier in that database for that particular taxon. So you don't have to care that your accepted name in one database is not the same as your accepted name in another database, so long as they have the same ID.
Yeah.
And so you just reconcile them all to the same taxonomic reference, and then you just work on those individual references.
Yeah.
Taxonomists don't like that much because effectively you're ignoring what the Latin name says and using the identifier for a taxon as a number effectively. But it's very difficult. I mean, probably most of you have tried reconciling taxon names in data sets so you can match things up, and it takes an awful lot of time, and there's no common list of accepted names for taxa in the world. It depends on the authorities you use, what they think, but that's quite a powerful way of doing it.
Yeah.
Sorry,
and here, for example, I can just call the GBIF taxon ID, and then I can take this ID, go to GBIF, and get a bunch of other stuff from there. But say, for example, I've got this data set which uses these Latin names, and then I've got a different data set that uses a different authority. If I match to GBIF taxon ID, I should be able to connect those two all the same.
Yeah, the GBIF taxonomic backbone is one of the best or most complete backbones we have for the world to reconcile against. However, there's lots of mistakes in it. But if you're doing big projects with thousands of species names, a few mistakes are not going to make much difference, probably. But you have to be aware there are mistakes in the backbone.
Just one question, Peter.
Yes.
So if you start to reconcile and add in new information, I think this is a really nice feature of this Open Refine. It always, I know it keeps track of all the edits you do, but how messy can it get? Because we added all those common names, and then if we add another one that has another extended bunch of information, so you end up having one column for your main entries, your number of original entries, and then you have, like, I don't know, like you said, multiple entries with no assigned information. It always keeps track of that, and you can always automatically refill everything, because in the end, no one wants to go just like fill this and refill this.
No, yeah, you can fill down automatically, but as long as you keep it on the record view here, it'll just keep track on its own, because it's using this field over here.
Okay.
That's the internal ID. So we've got this ID, which is a real column, but OpenRefine has its own ID as well, and it uses that. And
okay,
this
so
here,
you're working at the record level.
Yeah.
Okay.
But something else that I failed to mention is you've got like flags and stars here. You can use those in your workflow and use them for whatever you want. You can sort on them or filter on them. That's really for, like, maybe you use flags for issues. Maybe you use stars for things you need to return to. This is like a quality of life thing. And something else that I didn't mention is you can also sort, and you can use like pretty fancy sorting. Like, for example, if you used one of those Grell things I showed earlier, which one was it? This one, for example, for maybe if it's one of these two values, it should be true. You could use a sorting here to only get the true ones on top. I personally am not in the habit of sorting a lot in data at all, so that's why you see me using these facets all the time instead. But it's all up to your preference. I usually work on data sets that inherently have no order, so that's why I don't sort. And you'll see me often like hiding columns, like collapse this column. This column is actually still there. But that's just to make it easy for myself. I use OpenRefine really as when I want to do something quickly or explore a lot of data quickly, or when I want to connect to Wikidata or GBIF or whatever for true cleaning, I tend to step back to R. But I think OpenRefine is a valid option in many cases. Something that I'm not going to go into.
I think Nigel wants to.
Oh, sorry.
Yeah, I just had a question, and
I can't see everyone. Sorry.
Yeah, no, I didn't. Peter, how would you like report what you did with OpenRefine if you would clean the data set and then use it for further analysis and not go back into R to repeat that?
So the completely repeatable way of doing that is going to your edit history, clicking the extract button. You can remove everything here that you don't want to mention, and copying this and then publishing that alongside your data set as a JSON archive. And then maybe you also want to describe these things briefly as well.
Okay.
But the reason I showed you this one earlier with the 600, this is not by far not the most you can get up to. As with many things that involve manual checking, if you're going to report on every step, you're going to end up with a very long list. So it's useful that you can do this, but it's got limited utility in my opinion. But I guess it's still a lot better than just doing it all manually and not writing it down at all.
Yeah.
And at least it's keeping track for you. So maybe I'm changing my own mind here. Actually, it is quite useful. And also, I'm working on 260 records. If I make a change here, even though I can't see them, it's still doing them until the 260 are over. I just want to keep repeating that so you remember. About reconciliation. So I've got this connection with Wikidata now. I can also go the other way around. That's more of an advanced method that I'm not going to be demonstrating. But for example, if I have something in my data set that Wikidata doesn't know, like for example, I know which species are invasive and which are not, and Wikidata doesn't know. You should check in advance, by the way. You can also push that to Wikidata from OpenRefine. So you can create quick statements, and then if you're you've already used Wikidata a little bit, you can use them, or you can export them as Wikidata files, and I need to log in, and then I need to remember my passwords. Yeah, tough luck, sorry. Then it would allow you to make those changes automatically. But before you start doing that, you probably should, it's really good, but check if they're made before. Check if the information is not already there, because you can really do a lot of changes in Wikidata very easily with OpenRefine. But I think that's using OpenRefine to make changes to Wikidata is such a big topic that we should probably cover that in a different session, in my opinion, at least.
So, Peter, you've been going an hour, and thank you. That's a lot to do and a lot to get through. So normally speaking, the coding club goes for an hour, and so the coding club people might want to go. But I thought what we could possibly do, the training course goes on officially till 6:15, another hour and 15 minutes. But it's really up to people here what they want to do next. I thought if people do want to stay on, they could do some sort of hands-on stuff. I don't know if anybody installed OpenRefine. It's a very quick install if you have Java already on your machine.
And Maven.
Sorry?
And Apache Maven.
Didn't think I had that on there, but I installed it this afternoon and it worked fine. So, but I already had Java for sure.
Yeah. If anybody has something that springs to mind, like, could I use OpenRefine for this? Now would be a good moment to ask.
Yes, absolutely.
So if you feel ambitious, you can just tell me. For example, OpenRefine is not limited to the stuff we've shown here. Like you can do math in OpenRefine, you can do all sorts of fancy matching between columns. I've mostly gone from one column to the outside world, or told one column how I wanted it to look, but you can have these columns talk to each other and interact. And you briefly saw me make, you can do conditionals, like change this column into this if this is the case, or otherwise do this. You can also duplicate columns, which is really handy. You can connect to websites and get information from there, even if it's not a true database, but be careful, because if the website is inconsistent, then you'll get inconsistent results. That sort of thing.
I also saw that one of the things that you could import were databases, not simple files. Do you know anything about that?
I know it works quite well, but I don't actually have a database up and running, so I can't show you. So see, I don't have any.
Like, I don't have you're inputting single
Yeah.
tables, so it's still square data.
Yeah, but you can. The handy thing about connecting to a database is that even if it's really big, you can still import it. If you look at the projects that I have open, you can have a look at the biggest one. The biggest one is 1.7 million rows. Is it? Yeah, 1.7 million rows. So OpenRefine really works quicker and better for larger sets than you could reasonably edit in Excel. But to be honest, if you're working with sets this big, it's easy to make oversight mistakes. Something might be true for 99% of the cases, but if you've got 1.7 million rows, you're still making 1,000 errors. So maybe, so really go deep in the exploration and make sure that what you're doing is correct. And you can use the filters, the facets, to only do your manipulation on the ones where you know that what you're doing is correct.
One thing generally about data, and this is used a lot for things like georeferencing. So if you have a lot of specimens and you know the locality string that's been written on them, you can use that then to sort of put it a longitude and latitude on them. But what often happens with data is you'll have a lot of things from one place and a lot of places from one, so a lot of one-offs. So you have a curve where you have a load of specimens from London, and then a whole long tail of places where the place is just one specimen from everywhere. So Google Refine is great because of its sort of faceting and clustering to get those really common ones, and you can get those done really quick. But you're probably wasting your time if you're working on that tail, because effectively you're just doing one after another like you would in a spreadsheet anyway.
Yeah, but it's really useful for finding the tail. That's something that's not always trivial using other tools. And you saw me doing that earlier. Like I took everything that matched to Wikidata, and then I went to look like which are the taxa that Wikidata doesn't know, that really has no clue about, doesn't know the family, know nothing. And then I saw, oh, Wikidata doesn't match to the SP suffix. And if I keep doing that, I'll eventually end up with taxa that Wikidata just plain doesn't know about if my data set is big enough. And maybe you could add those to Wikidata, or you could ask yourself, is this really a taxa? Is there maybe the wrong value in the field? Is this like a spelling error? That sort of thing.
Okay. Is there anybody who wants to stay on and actually try it out themselves? Or I can see a few people from the coding club have left because they had other things to do. I know Peter can talk about OpenRefine for another hour if we let him, but
sorry,
that's absolutely fine. I think that
probably if I was if I had more of like a set lesson plan or a presentation, then I would have had more of a story to tell. But
I'm just aware that the trainees have been completely saturated with information today. And hopefully some of it sunk in, but we shouldn't push our luck.
So let me just share one more thing in the chat. It's on the OpenRefine GitHub, there is a list of resources and courses and tutorials on OpenRefine. just going by the titles, you can probably find some stuff that you want to learn about. So like if you're interested in fixing strings, or if you want to connect to databases or that sort of thing, you can probably find a topic in there. And there's also some really good general courses. I think data carpentry also has a really good general course.
But one thing I forgot, Sophia, is we never did a group photo.
We should do a group photo,
We should
right?
do a group photo.
Of course. Let's do a group photo.
So and we can still hang around if anyone has some last questions after this, but we can have a group photo and then and say goodbye to anyone who wants to leave, and then hang on for anybody who just wants to chat. So
can you please turn on your video? Who will do it? Quentin, do you?
How do we do this? It's so many people. How do we
And maybe the coding club ones remove so that you can have the ones who attended the course this week and have everyone inside.
It still fits on my screen.
We could do two. We can do the whole thing, and then we do just the people who the trainees.
Yeah.
Yeah.
So Helena, come back. We'll do the whole thing first. And maybe is it 2 pages? Yeah. So we're gonna have to. You're gonna do it, Joanna or Sophia,
or you?
Or me? Okay. Okay. So everyone say cheese.
Joaquin, Sabina, Paulina, can you turn on the cameras?
All volunteer.
Silvana, are you there?
But my camera is on. You cannot see me.
Yeah, mine is on too.
I can see you.
Ah, now I see you. Oh, probably the problem was with mine, not yours.
Just do it, Quentin.
Okay. I was just trying to take the participant list off because so I wasn't prepared. Okay. So cheese. Okay, and I need to just paste that somewhere. I'm going to have to do page 2. I hope we don't get too much overlap. Say cheese again. I'm going to be in both. So
it's just you and your twin brother. I've actually got my Zoom set up so I can see 50 people on one screen.
Oh, well, you should have been doing this, not me then.
Yeah, well, there you go, picture.
I think I just overwrote the wrong. You already done it, have you?
I did just now. Does everybody want to smile again?
Do it again, because I think I just overwrote the one I just did by accident. So cheese.
There you go.
Okay. And then all of us who aren't trainees turn off their cameras. Do you all still fit on the same page, I wonder? Okay. So cheese.
Got it?
I think so, yeah. I'm assuming it all went on one page that time, so. Good.
Funny moments. Well, thank you so much, Peter, for this lovely demonstration. It's much easier than to use the VBA in Excel to do all these type of processes. So thank you so much as well for sharing with us today. And Quentin, a special thank you to you for all these hours.
For all the organization and everything. My God, yes.
And of course, to all the participants and to the other people that joined us today afternoon. Well, you have our contacts, so just reach out to us for anything you need regarding the theme of the course. And yeah, don't forget to send us your final report for this course, the essay. And let's keep in touch.
Thank you.
Thank you so much.
Thank you for everything.
Thank you, Quentin, again.
Thank you.
Thank you. Thank you very much.
See ya.
Thank you.
Thank you. Bye.
Thank you.
Thank you.
Bye.
Bye bye.
Bye-bye. Bye-bye.
Bye.
Bye-bye.
Bye, Nicola.
Have a good weekend all together.
Yes.
Bye.
Thank you. Bye. So,
Já
Heliana,
saíste? Deixa-me sair, olha, valha-me, deixa-me dizer que eles ainda estão a falar.
So Quentin, I'm a bit lost now. Is the coding club now we ended, right?
if you wanted to do something now, talk about something, I'm not going anywhere or doing anything now. So if you want, looks like I can't see Peter anymore, so it looks like we can't do Open Refine.
Yeah. Well, I will try it. I'm installing it because I didn't have time yesterday, and I'll go through it. And if I have any doubt, then I'll let you know. But I will stay here for a while. I don't know if you still, maybe I'll.
I need to sort out these photographs of people so I don't lose that.
Oh, but I guess no one is here, right?
I think the rest have gone to sleep.
Marina.
Oh, there's Marina. Okay. Hi, you.
It's for your first.
How did you find it, Marina? Peter talks very quick.
It was a normal pace for me, like a good pace. Yeah, I was able to follow. I mean, I knew a little bit about it, like it wasn't completely new, but I've never really used it myself.
Yeah, what I really enjoyed it was the reconciliation thing.
Yeah,
that is really quick and fast way of getting new information. That is nice.
Yeah, but also finding hidden duplicates.
Yeah, that's. I don't know. Well, if you do that, and then you want to keep an audit, a really nice audit of your cleaning data for, like you said, one of the things like a workflow that is reproducible. You don't gain much of doing it here, right?
I was thinking you'd want to create a duplicate column, clean up one of the columns, and so you have the original or something like that still there. So you could, because if you wanted to match it back to the, yeah, I just don't like getting rid of the original very much.
Yeah, just thinking. I was all day listening to the talks. They were really interesting. And if you think of the don't touch a raw data principle, and you do these things for exploration, then you have a lot of work done. But you still have to code it if you want to replicate and have a steady workflow for other circumstances. Yeah. So it's a bit of a trade-off. But I think we can adapt to the needs. We probably don't need a workflow for everything, and this will still be very useful.
Yeah. So Marina, were you there this morning?
No, because I had 2 other meetings today.
It's fine. No, it was a really nice morning actually. But my brain is just about exploding with all the information that we got today. Plus Zoom is not as much fun as being face-to-face, that's for sure. So.
Yeah. I always have our lab meetings on Friday, and then so one of the 4 Fridays in a month, also another meeting at 2. So and then the coding club. So it's usually like 2 or 3 meetings on Friday.
Right. Well, now I've kind of committed to do coding clubs every week. But of course I'm going to have to juggle when I'm not around, and I can probably keep meetings out of this particular time slot, but like it doesn't always work. I was able to combine the 2 in this particular case, but that's kind of rare. But we'll see how it goes.
Quentin, can you send that Google sheet for the different sessions by email? Because I lost completely the link, and now I don't know where I have it. And you asked us for proposals.
So you have ideas?
Well, but we need, I think we need to make an effort to contribute. Otherwise, it's all on you. It's not.
But Liana, I think also if you just open, like if you type Google spreadsheets and go to your Google spreadsheets, it will be in the history, probably like the first one.
Because I have so many. I tried to do
called
it
Coding
the other
Club
day,
timetable, but I'm just sharing it with you now.
and I don't know where I couldn't find it. I was like, ugh. Oh, okay, I will keep these links.
And anybody internet can edit, apparently, so it's fully open.
And one more thing, Quentin, after the day of today, I have all the, I need to start, this coding club is really nice, but then it's really frustrating because you have all these ideas and then you just need to stick, focus and just start doing something.
That's my life. I'm full of ideas I can't do, so.
So yeah, after today, I had already seen some of your stuff in trials and from also other presentations, Leon's stuff, and I was really would like to try to implement the workflow because I have this data for this Nature 2000. It's where we are going to do the BioBlitz for the LNCSI next year, and I would really like to compare all this historical data because there was no record whatsoever for this on invasive species for this area on exotic non-indigenous species. And now we have this database that we have pulled together from historical literature, whatever we could find. And we have the data, the occurrences, all these things. So I would like to close it, publish it, have these historic records, and then be able to pick up some of these things and select the species for the BioBlitz, because I think some of them we found one record, we don't even know if they are here or there, so we'd like to target specific ones and see if with the BioBlitz we could increase at least a better notion of their distribution. But in order to do that, I need to close this database and publish it, like the workflow, everything, and then start bringing new data with the GBIF updates or the iNaturalist updates or the BioBlitzes. But I would like, how easy do you think it would be to run this trias workflow into a, historic database, which we don't know? And
the database you've created from things of lots of different places, is that just basically a spreadsheet?
It is. I have different. I collected a lot of information, and I was trying to see if there was something that I could build a database like open source, because we have so one spreadsheet has a lot of columns, and it's on the record. So the observations, the occurrences that we got. Another one is related with the species pathways of introduction and introduction history, this kind of thing. And the other is on the impacts, and that's a bit more complex, but it's also very interesting. And with that, I would like to do something more professional.
I'm also reminded, because Marina wants to publish data from Ukraine.
Yes.
In a very similar way, don't you?
I do, yes, but I'm not even nearly done with that.
But I mean,
I was telling you, it's like hundreds, thousands of data sources.
I also have the Portuguese one. So we had checklists, but no one had gone through each of the papers and picked the location. And if they didn't have a georeference, because back then they didn't, we would go back to Google Earth to the description of the paper and georeferenced it exactly where they say. So below the pier in beach. So we have all those georeferences that weren't georeferenced before. So I think it's really interesting, and I would like to have that. So we have more information for our local study site, but I have for all Portuguese coastal. So, and I think it's a pity. I just want to get rid of that.
I think, I don't think it's that difficult. I think we should talk to Lynn about it, because I think it would interest her, because she would love someone to use her workflows. And for some of it's so you've got observation data. So a lot of what Lynn did, she was talking about today, was checklist data. So so it's like checklist for the whole country. But she's also published observation data in a similar way. And it sounds like all we need to do is really just map one onto the other.
Okay.
And for Marina, I mean, if you take the whole Ukraine, that's a lot. But maybe you want to just take chunks of it, publish just bits of it at a time to GBIF, and so grow it slowly in a more manageable way.
Yeah, I don't know. I mean, always it's like always conflict of interest. Maybe it's Ukrainian mentality, but I'm sure that like my colleagues in Ukraine, if they see me doing that, they would be like, oh, why are you doing this and not us who are actually in Ukraine?
Yeah, so I mean, we have the similar sorts of things in, well, it's not quite exactly the same in Belgium, but it's really trying to pitch it to them as you're going to help them publish their data. So you say to them, I've seen your data. It would be really great if it was available on GBIF for everyone to use. Would you like me to help you publish it? And maybe that's a way to do it. I don't think it'll work for everybody, but maybe you just need to get one person for a start. And then and then when you put it onto GBIF, the data set has their name on it.
Yeah, the thing is that I tried like 2 years ago, I tried setting up a small team of people who are, well, botanists who work a lot on non-native species. And either they didn't understand the whole thing. I mean, they are like, don't really work with data much, and maybe they didn't get the point of what I wanted from them. I don't know, but it didn't work out.
Could we, I mean, Ukraine is a cost associated country or something, isn't it?
Yes, it is.
So couldn't we ask Helen for some money to run a data publication workshop in Ukraine?
Well, I think there were a couple, so we have, I know some people in Ukraine who are these GBIF ambassadors.
Right, yeah, exactly.
And they have ran some workshops. So there is like a group of people who are familiar how to do all these old things, and they are trying to mobilize some data from Ukraine.
But we can invite all of those people too.
That's the thing. I don't like the
engagement too.
You don't like
I have
those
I have a personal.
Okay.
I don't. So in Ukraine, we're also trying to
Then we won't
create.
invite those people.
Like, no, the thing is, like, what they are like, I'm a user of GBIF, and I think that all the data needs to be open, but I also see the value in creating something Ukrainian, because Ukraine is still like, has this like very secluded, I don't know, worldview. So we need to start with something smaller. And now these people who were appointed as GBIF ambassadors, they are doing things against the people who want to do something internally in Ukraine, which is like, sounds very weird,
We should all be
but it all
together.
goes like in all this like gossip, and they have been spreading bad things. Like, I know this like from inside, because I was helping the Ukrainians who were creating the Ukrainian database and portal for biodiversity data. So yes, I don't want to interact with those GBIF ambassadors in Ukraine.
Well, that's sad. This is the thing, but.
But do you need to go through them? That's my question.
No, like,
because
they are, and especially since
Yeah.
I am interested in plants in particular, I know many people who are just botanists or plant ecologists who have a lot of data, and I'm sure that they are like, they don't really see the full potential of the data that they have.
I know that different countries have their own inside platform of GBIF, because when I first registered, it was through inside of iNaturalist.
Yeah, but for that you need, your government needs to sign an agreement. Like, you need to become a.
Okay.
Well, you can be like a voting member, or just but then you need to pay for membership. And in Ukraine, we were trying to do that. Like, we prepared an official letter that then was sent from the GBIF to the Ukrainian prime minister, I think it was, and they just ignored it. Nothing happened.
Yeah, even in rich countries, I mean, big countries like the USA are usually 2 or 3 years behind with their payments and things like this. It's always a struggle for this kind of thing. So they talk a lot about helping biodiversity, but actually when it comes to it, and GBIF is not expensive for a country.
Yeah, absolutely.
So so getting back to these data sets, I at lunchtime wrote to Helen and said, we need to have a Alien CSI hackathon So that could be a way that the whole coding club and the trainees here could get together sometime next year. And I'm thinking in springtime, not too soon, and keep my fingers crossed that we finally can put this pandemic behind us.
Yeah.
That we can actually get together and do sort of data publication, try playing around with the APIs for iNaturalist and stuff like that. I don't know what we'll do. It's a hackathon. We just hack. I'm sure Nigel will do some amazing ggplot and and what have you. And so I'm assuming we can do stuff like helping Heliana publish her data set and things like that. Get Lynn in the room with Heliana. The other way to do it is to do it through short-term scientific missions.
Yeah, I never applied because they are for young researchers. I passed the young researchers.
They're for you too.
And the young researchers in invasion ecology, if that counts, not in researching.
There's STSM, and none of that acronym is young in there. There's no Y.
Yeah, we could do, because I really would like to have, split the historical data sets because they come from papers and theses, whatever, just have that closed and then start to play with the new platforms, because and find a way to integrate them, because I think more and more.
Tell you what, 1 year, well, no, Lynn has 2 kids. I was thinking you could invite Lynn to Portugal for 2 weeks and she'd drop everything and come, I think.
She could stay here with the kids.
So, but maybe you have to come to Brussels and spend her time here, because I'm not sure she'd leave her kids for 2 weeks. So
that would be a nice idea. We could apply for a STSM. I don't know if they will open now again in October. No, something, isn't it?
Well, so I think because it's all got messed up. So I think the current year finishes in the beginning of October, then it takes a month or 2 for the next budget to sort of kick in. So it'll still take a while before they have their next SCSM call. But it's a short year, so I guess they should hurry up about it.
Yeah.
But I don't really know what's going to happen exactly, whether they extend it or what, I don't know.
That would be nice. We could refine that to the occurrences, integrate with the work she has done, and then prepare it to link the data sets on the trend with the BioBlitz that will happen in May in the same area. That would be perfect.
Exactly.
Yeah. Because it's when they were talking this morning about the use of mixed data, we always have to use mixed data, particularly in these sites where we don't have a lot of funding. We cannot afford not discard this data or that data. If I have it, I will.
You'll use it.
Yeah. So, yeah. Okay. I'll take notice when they open again. We could prepare something. That would be nice. Go and spend.
Heliana, which city are you in?
Marina? Me or Marina?
You.
Heliana.
In Aveiro. You know where it is?
No.
South of Oporto, in the coast.
Oh, okay.
It's like
it was just in September. I go hoping to go to I can't pronounce it. Coimbra.
Oh yes, that's where I studied. Yeah, it's also very near here. It's like 1 hour from here.
Okay.
Inside.
Well,
when do you come here?
September 8th, 9th, 10th, I think.
It's a meeting.
Hopefully a workshop on using machine learning in collections.
Okay. It will be in the Botanic Museum or garden or something. Yeah. Nice. Maybe we can arrange something. I can meet you there. We can visit here. Yeah, sure. It's been nice. I'm probably here.
So Sophie Mers was telling me there's a group that works on Oxalis pescapri somewhere there, but maybe it's in Coimbra. How do you pronounce it? Coimbra?
Coimbra, yeah.
Coimbra, okay.
Yeah.
But I don't know where that exactly is. But we went through, actually we got a student to go through iNaturalist records. So Oxalis pescapri is one of these heterostylus plants. It has long, medium, and short styles.
Yeah.
And so we got a student to go through looking at the pictures on iNaturalist and determining whether they had long, short, or medium styles for every. everywhere in its invaded and non-invaded and native range and things.
So the most common, because I remember you talking about this in Kratiri, the most common one are the short ones.
Oh, you got a good memory.
Yeah. So I remembered after a while, I came here and every time I saw one, and it was always
Short style one, yeah.
And then it became a bit boring because there are no others here. I haven't seen.
No, they're definitely in Portugal. Portugal, outside its native range, Portugal has got the most long-styled ones.
Okay, now you got my interest again. Now I will check. I will send you pictures if I find.
So what's interesting is that there was a group from Portugal who they had about 50% long-styled populations and 50% short-styled, and there is some hybridization between the two and a few medium, middle-styled ones. But when we took our naturalist and then looked at all the Portugal ones, we find very few long-styled ones in there. There are just a few, but more than most places. But what's obvious is the people who did the study in Portugal were picking their populations basically, because they had so many long-styled ones. And the nice thing about
where those occur, it's spread along the country.
It's all mostly up and down the coast, but it's not really in northern Portugal. It's mostly from sort of 3/4 of the way down around the coast and along the Algarve and things like that.
I thought the long ones, you say?
Oh, they're scattered around. I mean, most of the dispersal is via the bulbs still, rather than the seeds.
Because you find them everywhere here. I remember seeing them ever since. So.
And the double-flowered ones are kind of interesting too, because they're not. The double-flowered ones, again, people spread them around from horticulture, but they're not everywhere. So in California, they're almost exclusively short-styled ones. In New Zealand, there are long-styled ones. And so it's tracking the introduction history of these things. It's not quite logical like you might think.
Okay, that's not interesting. No, you do let me know if you come here in September. It would be nice to meet. Yeah. Okay. I will play a bit with this. See, I will also follow the Darwin core to update the field names of our database because we built it before it was approved that alien species more.
I see Nadia has put in, she's going to talk to us about Biomod 2 next week.
Ah, nice. Distribution modeling.
Exactly. Yeah. So that's good.
Yeah.
I wonder if you learned about that this week.
Yeah, I know they use some models I saw in the program. I did a course once, which was nice on SDM. It's another package, which is also very nice. I tried to work with the biomod once, but then I didn't follow through.
Just a few, but can't remember what I've done last time. It may have been Biomod I used last time.
Biomod 2 and Sesam R. They have Sesam R. And this is new. I never heard of it. Yeah.
But if you want to talk about other things, like I had a list, I think I already shared it with you, of different subjects around coding, which are not necessarily about coding specifically, then I'm quite happy to have an hour talking about those things too.
Okay.
Like how to approach people amateurs about data in Ukraine or something like that. Who to avoid in Ukraine.
I mean, I didn't mean to put you against any. It's just
no, I'm just pulling your leg. That's fine.
Yeah.
I'm not a person who usually gossips about other people. They did some pretty nasty things. So
really?
that's why.
Yeah, that's just silly.
So you should go and publish the data through international.
No, those are not the people who own the data.
Okay.
These are
the ones that manage
people who do other. Yes,
yeah.
They basically stole a grant for which we wrote the proposal.
Oh gosh, that's bad.
That we got from the Dutch from NLDIF, and yeah. I mean, that was just like that. So we got a grant to develop to upgrade the website for the Ukrainian biodiversity database, and install API and then link to GBIF. And they took the, I mean, like using some internal contacts or like some lies, they took it from, they persuaded the manager of the grant, who's the Dutch person, not to give money to us, like to like, I mean, that was just so bad.
Yeah, that hurts. Anyway. Yeah.
Anyway, sorry.
No, it's fine.
Maybe I shouldn't have even.
I have all sorts of stories about painful things in science where people have stabbed me in the back. So fortunately, not too recently, but I know how bad it hurts, to be honest. So, okay.
I'm just trying to find the list.
The list of things I suggested. Yeah. I can't think where it is either.
It's the link you put in the chat right now, right?
No.
And that's the list of the clothing.
In the email?
That was in the email, yes. The list of clothes.
But do you have that email? Because I can't remember what
I still have it. You needed me to forward it to you?
I was just trying to, well, we could put that in another tab and then it'll remind me what the other things we were going to talk about are.
Let me open it.
Because even though I listed them, I'm not very competent to talk about all those subjects.
I have it somewhere in here. So clothing club. Okay, so I will put it in the chat, all these topics that you listed. There it is.
All right. Okay. I'm going to now cut and paste them into the other tab. So, or the other, yeah, we need a new tab. Are you going on a holiday at all this summer?
Yes, I will try to take my holidays in August this year because I have work until the 28th, so I can do it before that, of July, but not the entire month. Still haven't decided quite well. But I'll probably be attending the sessions unless I'm in some beach with no internet, which is not very likely this year. Let's see. Otherwise.
Yeah.
Yeah, I will go home, well, to visit my parents in Ukraine finally after almost 2 years.
Oh gosh.
And then have a vacation with my husband, whom I also haven't seen since Christmas because he lives in London.
And where are you, Marina?
I'm in Halle, so it's near Leipzig in Germany.
Okay, in Germany.
And he hasn't been allowed to enter Germany because of the Delta.
Yeah. No, my parents, obviously, and my family are in the UK, and so they changed the rules just in the last few days.
Yeah.
If you're the rules, it said at first that if you're vaccinated, you don't have to quarantine. It's only if you're vaccinated by the NHM, the National Health Service in the UK. So most expats like myself are vaccinated in Europe. We still have to quarantine. It's just stupid. It's the same vaccine.
Yeah, it's really stupid.
Yeah.
Oh, really? I didn't realize that.
Yeah. So you still may have to quarantine.
Oh. But we need to, but we are increasing restrictions. For example, tomorrow I was going to an exhibition of a friend of mine, is a photographer in Oporto. And we were planning to have dinner because I haven't seen them for a while. And this week they already warned that those who don't have the second doses of the vaccine won't be able to have dinner in restaurants inside. So that's so much for our dinner already. And I'm just hoping they don't close the inter-municipality movements. And at 11, I have to be at home because now it's already... how do you say curfew?
Curfew.
So I guess I think it's not stopping this thing. And I already have one of those.
We've managed to have this workshop next week for the BioBlitz paper, and there's 6 of us who've managed to get together. 3, Nikki from Cyprus, Christina from Romania, and Bernat from Spain. +3 Tim as well from Belgium, and Sophie from my institution as well. So there's 6 of us. So we're actually going to have a face-to-face meeting, and we're going to try and go out for dinner. But my God, it's not easy.
No, it's not. Well, you're 6. The limit here is 10 now.
All right.
So
we're trying to organize a hackathon in September, but this is a kind of a trial run because we're trying to do a hybrid event. So there's 6 of us on site, plus we're going to have lots of virtual meetings. I think we're going to be exhausted by the end of the 3 days because trying to keep track of all the virtual people as well as the people on site.
Yeah, it is
easy,
challenging.
Anyway,
okay,
we're learning. Okay, well, I guess I should say goodbye now. And
yeah, we see you next week. Thank you, Quentin, for the link.
Thank you for being
here.
here.
Thank you very much. Bye-bye.
Bye.